So that being said, I'm going to hand it off to our chair, Dr. Monto. Dr. Monto, are you ready?
I'm ready. And it's my pleasure to open the second day of the 174th meeting of the Vaccines and Related Biological Products Advisory Committee. Our topic for today, and we have a double topic. The committee will meet in open session to discuss amending the EUA of the Moderna COVID-19 vaccine to include the prevention of COVID-19 in infants and children 6 months through 5 years of age. And the second topic, also to discuss amending the EUA of Pfizer-BioNTech COVID-19 vaccine to include the prevention of COVID-19 in infants and children 6 months through 4 years of age. We next will have Prabha Atreya, the designated federal officer, open the meeting on her side, introduce the members, and go through the housekeeping issues. Over to you, Prabha.
Thank you, Dr. Monto. Good morning, everyone. This is Prabha Atreya, and I'm the designated federal officer for this 174th Vaccines and Related Biological Products Advisory Committee meeting. And it is my great honor to serve as the DFO for this meeting. On behalf of the FDA, the Center for Biologics Evaluation and Research, and also the Vaccines Advisory Committee, I'm very happy to welcome everyone for today's virtual meeting. Today, the committee will meet in open session to discuss amending the emergency use authorization of the Moderna COVID-19 mRNA vaccine to include the administration of the primary series to infants and children through 6 months through 5 years of age, and also to discuss amending the emergency use authorization of the Pfizer-BioNTech COVID mRNA vaccine to include the administration of the primary series to infants and children 6 months through 4 years of age. Today's meeting and the topic were announced in the Federal Register notice that was published on May 31, 2022. At this time, I would like to introduce and acknowledge the excellent contributions of the staff and the great team I have in my division in preparing for today's meeting. Dr. Susan Pedar is my alternate DFO who will read the conflicts of interest statement for the public record. Ms. Christina Ward, my backup DFO, will be conducting the voting process later today. In addition to Susan and Christina, the other staff who contributed significantly are Ms. Joanne Lipkind, and Ms. Karen Thomas, Ms. Lisa Wheeler, and Ms. Viola Sampson for this meeting. I would also like to express our sincere appreciation to Mike Kaczynski in facilitating today's meeting. Also, our sincere gratitude goes to many CBER and FDA staff working hard behind the scenes, trying to ensure that today's virtual meeting will also be a very successful one, like all the previous Vaccines Advisory Committee meetings. Please direct any press and our media questions for today's meeting to FDA's Office of the Media Affairs at fdaoma@fda.hhs.gov. The transcriptionist for today's meeting is Ora Giles. We'll begin with today's meeting by taking a formal roll call for the committee members and the temporary members. When it is your turn, please turn on your video camera, unmute, and state your first and last name. And when finished, you can turn your camera off so we can proceed to the next person. Please see the meeting member roster slides, in which we will begin with the chair, Dr. Arnold Monto. Dr. Monto, can you start, please?
Yes, thank you, Prabha. I'm Arnold Monto. I'm at the University of Michigan School of Public Health, where I have been involved in research on prevention and control of respiratory infections, flu, and COVID-19 for a number of years. Back to you, Prabha.
Thank you. Next, Dr. Paula Annunziato, who will be joining a few minutes later, and we can proceed with Dr. Adam Berger.
Hi, I'm Adam Berger. I'm a geneticist by training. I'm also the Director of Clinical and Healthcare Research Policy at the National Institutes of Health, where I oversee all of our clinical research and clinical trial policies. Thanks.
Thank you. Next, Dr. Hank Bernstein.
Good morning. I'm Hank Bernstein. I'm a professor of pediatrics at the Zucker School of Medicine at Hofstra and Northwell. I'm a general pediatrician with special interests in vaccines and public health.
Thank you. Next is Dr. Archana Chatterjee.
Good morning. My name is Archana Chatterjee. I have the privilege to serve as the Dean of Chicago Medical School and Vice President for Medical Affairs at Rosalind Franklin University in North Chicago. I'm a pediatric infectious diseases specialist with expertise in the field of vaccines. Thank you.
Thank you. Next is Captain Amanda Cohn.
Good morning. I'm Amanda Cohn. I'm a pediatrician and public health expert at the Centers for Disease Control and Prevention with expertise in vaccine preventable diseases. Thanks.
Thank you. Next is Dr. Offit, Paul Offit.
Good morning. I'm Paul Offit. I'm an attending physician in the Division of Infectious Diseases at the Children's Hospital of Philadelphia and a professor of pediatrics at the University of Pennsylvania School of Medicine. My area of research interest is mucosal vaccines. Thank you.
Thank you. Next is Dr. Steve Porgum.
Thanks, Prabha. I'm Steve Pergam. I'm a adult infectious disease physician at the Fred Hutchinson Cancer Center in Seattle, Washington, and primarily focus on infections in immunosuppressed patients.
Thank you. Next is Dr. Jay Portnoy.
Good morning. I'm Jay Portnoy. I'm a professor of pediatrics at the University of Missouri, Kansas City School of Medicine. I'm an allergist immunologist in the Division of Allergy and Clinical Immunology at Children's Mercy Hospital in Kansas City.
Thank you. Next is Dr. Eric Rubin.
Dr. Rubin is running a little late, so we're going to move on to the next one, Prabha.
Okay, so next we introduce our temporary voting members, Dr. Vovita Fuller.
Good morning. I'm Avita Fuller. I'm in the microbiology and immunology department at the University of Michigan Medical School. I'm a virologist who studied viral entry and community engagement.
Thank you. Next is Dr. James Hildreth.
Good morning. I'm James Hildreth, the President and CEO of Meharry Medical College, Professor of Internal Medicine. I'm an immunologist and I study viral pathogenesis. Thank you.
Thank you. Next is Dr. Janet Lee.
Good morning. My name is Jeanette Lee. I'm a professor of biostatistics and a member of the Winthrop P. Rockefeller Cancer Institute at the University of Arkansas for Medical Sciences. Thank you.
Thank you. Next is Dr. Ofer Levy.
Hi, good morning. My name is Dr. Ofer Levy. I'm a physician scientist and attending physician in pediatric infectious diseases at Boston Children's Hospital, where I direct the Precision Vaccines Program, bringing precision medicine principles to vaccine research. I'm also professor of pediatrics at Harvard Medical School. Thank you.
Thank you, Dr. Levy. Next is Dr. Wayne Marasco.
Good morning. My name is Wayne Marasco. I'm a professor of medicine at Dana-Farber Cancer Institute and Harvard Medical School. I'm a practicing adult infectious disease expert. I'm also a research scientist, and the work that I specialize in is antiviral immunity and vaccine responses. Thank you.
Thank you. Next is Dr. Pamela McInnes.
Good morning. I'm Pamela McInnes. I am a retired Deputy Director of the National Center for Advancing Translational Sciences, one of the U.S. National Institutes of Health institutes.
Thank you. Dr. Meisner, Cody Meisner.
Just one sec. And Prabha, we also forgot a member we have to go back to. I just thought I'd share that with you. So.
Okay.
Go ahead, Dr. Go ahead, Dr. Meisner.
We are getting a lot of background, Mike.
Yep, I took care of it.
Okay, thank you. Go ahead, Dr. Meisner. We can't hear you. You are muted, I think.
I got it.
Thank you, Mike, and thank you, Prabha, and good morning to everyone. My name is Cody Meisner. I'm a professor of pediatrics and pediatric infectious disease at Tufts University School of Medicine. The Children's Hospital will soon close, and I will have a new address, but I appreciate the opportunity to participate in this meeting this morning. Thank you.
Thank you. Next is Dr. Michael Nelson.
Thank you, Dr. Atreya. I'm an allergist immunologist. I'm professor of medicine and chief of the Division of Asthma, Allergy, and Immunology at the University of Virginia. I also serve as the president of the American Board of Allergy and Immunology. Thank you.
Thank you so much. Dr. Art Reingold.
Good morning. My name is Art Reingold. I'm an infectious disease epidemiologist at the School of Public Health at the University of California, Berkeley.
Thank you. Next is Dr. Mark Sawyer.
Good morning. This is Mark Sawyer. I'm a professor of pediatric infectious disease at University of California, San Diego, and Rady Children's Hospital. My expertise is in the area of public health implementation of vaccine policy.
Thank you, Dr. Sawyer. Last but not least, Dr. Melinda Wharton.
Good morning. I'm Melinda Wharton. I'm an adult infectious disease physician by training, and I work in vaccine policy at the Centers for Disease Control and Prevention.
Thank you. Thank you, Dr. Wharton. Now I will call Dr. Susan Peidar to read the concepts of interest statement for the public record. Thank you.
Prabha, we have one more. Prabha, we have one more member. Dr. Gans.
Okay. If she's joined. Thank you. Sorry, Dr. Gans. So go ahead, please.
Thank you. This is Dr. Haley Gans, pediatric infectious disease at Stanford University. Relevant to our conversation today, my research focuses on the immune response to vaccines in both immunocompetent and also those children with suppressed immune systems, and I sit on many committees looking at adverse events. Thank you.
Thank you, Dr. Gans. Okay, Mike, do we have anybody else joining now?
No, we're good now. Thank you.
Okay, thank you. So, Susan, go ahead, please, and read your concept of interest statement for the public record.
Good morning, everyone. My name is Susan Peidar. It is my honor and pleasure to serve as the alternate designated federal officer for today's VRPAC meeting. Thank you for your attention as I proceed with reading the FDA Conflict of Interest Disclosure Statement for the public record. The Food and Drug Administration, FDA, is convening virtually today, June 15, 2022, the 174th meeting of the Vaccines and Related Biological Products Advisory Committee, VRPAC, under the authority of the Federal Advisory Committee Act, FACA of 1972. Dr. Arnold Monto is serving as the acting voting chair for today's meeting. Today on June 15, 2022, under Topic 2, the committee will meet in open session to discuss amending the EUA of the Moderna COVID-19 mRNA vaccine to include the administration of the primary series to infants and children 6 months through 5 years of age, and to discuss amending the EUA of the Pfizer-BioNTech COVID-19 mRNA vaccine to include the administration of the primary series to infants and children 6 months through 4 years of age. This topic is determined to be a particular matter involving specific parties, PMISP. With the exception of industry representative member, all standing and temporary voting members of the VRPAC are appointed special government employees, SGEs, or regular government employees, RGEs, from other agencies and are subject to federal conflict of interest laws and regulations. The following information on the status of this committee's compliance with federal ethics and conflict of interest laws, including but not limited to 18 U.S.C. Section 208, is being provided to participants in today's meeting and to the public. Related to the discussions of this meeting, all members, RGE and SGE consultants of this committee have been screened for potential financial conflicts of interest of their own, as well as those imputed to them, including those of their spouse or minor children, and for the purposes of 18 U.S. Code 208, their employers. These interests may include investments, consulting, expert witness testimony, contracts and grants, cooperative research and development agreements, CRADAS, teaching, speaking, writing, patents and royalties, and primary employment. These may include interests that are current or under negotiation. FDA has determined that all members of this advisory committee, both regular and temporary members, are in compliance with federal ethics and conflict of interest laws. Under 18 U.S.C. Section 208, Congress has authorized FDA to grant waivers to special government employees and regular government employees who have financial conflicts of interest when it is determined that the agency's need for special government employees' services outweighs the potential for a conflict of interest created by the financial interest involved, or when the interest of a regular government employee is not so substantial as to be deemed likely to affect the integrity of the services which the government may expect from the employee. Based on today's agenda and all financial interests reported by committee members and consultants, there have been one conflict of interest waiver issued under 18 U.S. Code 208 in connection with this meeting. We have the following consultants serving as temporary voting members: Dr. Ovida Fuller, Dr. James Hildreth, Dr. Jeanette Lee, Dr. Ofer Levy, Dr. Wayne Marasco, Dr. Pamela McInnes, Dr. Cody Meisner, Dr. Michael Nelson, Dr. Art Reingold, Dr. Mark Sawyer, and Dr. Melinda Wharton. Among these consultants, Dr. James Hildreth, a special government employee, has been issued a waiver for his participation in today's meeting. The waiver was posted on the FDA website for public disclosure. Dr. Paul Annunziato of Merck will serve as the industry representative for today's meeting. Industry representatives are not appointed as special government employees and serve as non-voting members of the committee. Industry representatives act on behalf of all regulated industry and bring general industry perspectives to the committee. Dr. Jay Portnoy is serving as the consumer representative for this committee. Consumer representatives are appointed special government employees and are screened and cleared prior to their participation in the meeting. They are voting members of the committee. FDA encourages all meeting participants, including open public hearing speakers, to advise the committee of any financial relationships that they may have with any affected firms, its products, and if known, its direct competitors. We would like to remind standing and temporary members that if the discussions involve any other products or firms not already on the agenda for which an FDA participant has a personal or imputed financial interest, the participants need to inform the DFO and exclude themselves from the discussion, and their exclusion will be noted for the record. This concludes my reading of the conflicts of interest statement for the public record. At this time, I would like to hand over the meeting to our chair, Dr. Monto. Thank you so much. Dr. Monto.
Thank you. And now I'd like to call on Dr. Peter Marks, the director of the Center for Biologics Evaluation and Research of FDA, to give us his welcome and tell us a little bit about what we are expected to do today.
Thanks very much, Dr. Monto. First of all, welcome to people who are tuning in, and thank you to both the advisory committee staff, the advisors, and to the FDA staff and to the sponsors as well as the open public hearing speakers for joining today. We appreciate everyone's participation. Today, we will be considering applications for amending emergency use authorization for both the Moderna and Pfizer BioNTech vaccines to include the younger pediatric populations of 6 through 5 and 6 through 4 years, respectively. If I could have the slide, Mike, that I sent you, just to remind people of why we're here. It's because even though there is a very high seroprevalence rate of the SARS coronavirus 2 in the pediatric population, there still was, during the Omicron wave, a relatively high rate of hospitalization during this period. If one looks at that gray period there towards the right of this slide, that was the Omicron period, and you can see a very sharp wave. That rate of hospitalization actually is quite troubling. And if we compare this to what we see in a terrible influenza season, it is worse. And the same way as the number of deaths in the 0 to 4 age range during the 2 years of the pandemic in total, as of May 28th, as we were reminded of yesterday by our CDC colleagues, the total number of deaths as of May 28th was 442 in the under 4 age range. That also compares quite terribly to what we've seen with influenza in the past. If one goes back to the H1N1 influenza season of 2009-2010, the number of deaths in that age range reported was 78, and we considered that pretty terrible. So we are dealing with an issue where I think we have to be careful that we don't become numb to the number of pediatric deaths because of the overwhelming number of older deaths here. Every life is important, and vaccine-preventable deaths are ones that we would like to try to do something about. We routinely give influenza vaccine across a broad age spectrum in order to help prevent deaths in precisely this kind of manner. So I just wanted to set the context here that the intervention we're talking about here is one that is something that we have accepted in the past to try to prevent deaths from influenza. Here we have a different pathogen, but one that has created a lot of havoc just the same. And so as we move today, I think we can kind of help, just wanted to help frame this in terms of the magnitude of the issue of COVID-19 in the youngest population. Granted, it's a population that has been much less affected than the older populations, particularly the oldest population, but one nonetheless has also been affected. I think for those who have lost children to COVID-19, our hearts go out to them because these are, each child that's lost is essentially fractures a family. So with that said, we'll look forward to, I think, a very good series of presentations, some excellent discussion, and wish everyone a very successful meeting today. Thank you. I'll turn it back to Dr. Monto.
Thank you, Dr. Marks. You've set the scene for what our obligation is today to look at the problem in the youngest of our population and to keep them as protected as possible using available vaccines. Now I'd like to turn the floor over to Dr. Sudhakar Agihotram from FDA. He is going to walk us through the agenda, tell us what we are going to be doing today specifically, and what the voting questions are going to be. This is a little unusual kind of meeting where we are looking at two different products, and he'll tell us how we're going to be managing going in and out of each with our discussions and then our voting questions. Sudhakar.
Thank you very much, Dr. Monto. Good morning, everyone. Can you all hear me? Okay.
Yes, sir. Go ahead.
Okay. Thanks very much, Mike. Good morning, everyone, and welcome to the second day of the advisory committee meeting for discussing the pediatric EUAs. Just a quick thing. This would be a co-presentation with Dr. Ramachandran Nayak, committee chair for Pfizer-BioNTech COVID-19 vaccine EUA request. So I will be co-presenting this presentation with Dr. Ramachandran Nayak. So with that said, I'm Sudhakar Agnihotram, primary reviewer and committee chair for Moderna COVID-19 vaccine EUA amendment. And I would like to begin my talk by thanking the FDA EUA Review Committee for Moderna COVID-19 vaccine, supervisors, management, for all their hard work that went into this, and also the advisory committee for their time in this valuable discussion. I would be providing an overview of the request from Moderna on amending their EUA for use of Moderna COVID-19 vaccine as a two-dose primary series in children 6 months through 5 years of age. Here is the outline of my talk, and then I will be providing a refresher on the currently available COVID-19 vaccine for use for primary vaccination in children. This would be followed by an overview of the request from Moderna on amending their EUA for use of Moderna COVID-19 vaccine as a two-dose primary series in children 6 months through 5 years of age, and the clinical package that supports this EUA request. Then I would be handing over the presentation to Dr. Ramachandran Nayak, and he would be talking about the overview of the request from BioNTech Manufacturing GmbH on amending their EUA for use of Pfizer-BioNTech COVID-19 vaccine as a three-dose primary series in individuals 6 months through 4 years of age, and the clinical package that supports this EUA request. He will also be providing a refresher on the statutory requirements for emergency use authorization, and Dr. Nayak will also provide an overview of today's agenda, followed by a presentation of the voting questions for both the Moderna COVID-19 vaccine and the Pfizer-BioNTech COVID-19 vaccine EUA requests. To give an overview of the currently available COVID-19 vaccine for primary vaccination in pediatric population, Pfizer-BioNTech COVID-19 vaccine is available under the EUA for use as a two-dose primary series given 3 weeks apart in individuals 5 years of age and older. Pfizer-BioNTech COVID-19 vaccine is also available under the EUA for use as a third primary series dose given at least 28 days after the second dose in individuals 5 years of age and older who have been determined to have certain kinds of immunocompromise. Comirnaty is FDA approved for use as a two-dose primary series in individuals 16 years of age and older and can be used interchangeably with Pfizer-BioNTech COVID-19 vaccine as currently authorized. To provide an overview of the request from Moderna for amending their EUA for use of Moderna COVID-19 vaccine as a two-dose primary series in individuals 6 through 5 years of age. Moderna submitted this amendment request on April 28, 2022, and then the proposed dosing and regimen includes a primary series of 2 doses, 0.25 ml each, containing 25 micrograms of mRNA given 1 month apart, administered intramuscularly in individuals 6 months through 5 years of age. The clinical package that supports this EUA request includes safety, efficacy, and immunogenicity data from approximately 1,800 vaccine recipients in children 6 to 23 months of age and approximately 3,000 vaccine recipients in children 2 through 5 years of age. You will be hearing a breakdown and much detailed presentation on this from both the FDA and the sponsors today. I will hand it over to Dr. Ramachandran Nayak now. Thank you very much.
Thank you, Dr. Agnihotram. Good morning, everyone. So, my name is Ramachandran Nayak from the Division of Vaccines and Related Product Applications in the Office of Vaccines and I am the review committee chair for this EUA amendment. I'm going to provide the background regarding Pfizer-BioNTech's EUA amendment request for the Pfizer-BioNTech COVID-19 vaccine for use in children 6 months through 4 years of age. Pfizer submitted the EUA amendment request on May 27th. The Pfizer-BioNTech COVID-19 vaccine is proposed to be administered as a primary series of 3 doses, 0.2 ml each dose containing 3 micrograms mRNA. First 2 doses administered 3 weeks apart, followed by a third dose administered at least 8 weeks after the second dose, administered intramuscularly in individuals 6 months through 4 years of age. The clinical data package includes safety and effectiveness data from about 3,000 vaccine recipients 6 months through 4 years of age. Details on this data will be provided in the later presentations by Pfizer and the FDA. As today's meeting is about discussion of amending emergency use authorizations of the COVID-19 vaccines, I'm going to reiterate the statutory requirements for issuing an EUA. FDA may issue an EUA of an unapproved medical product following an EUA declaration by the Secretary of the U.S. Department of HHS if the following statutory requirements are met. The agent referred to in the EUA declaration can cause a serious or life-threatening disease or condition. The medical product may be effective to prevent, diagnose, or treat serious or life-threatening condition caused by the agent. The known and potential benefits of the product outweigh the known and potential risks of the product. No adequate, approved, and available alternative to the product for diagnosing, preventing, or treating the disease or condition. Next slide is about the overview of today's agenda. After this FDA introduction, Moderna will provide the sponsor presentation, followed by FDA presentation by Dr. Robin Wish on FDA review of effectiveness and safety of Moderna COVID-19 vaccine in infants and children 6 months through 5 years of age. After the 15 minutes break, Pfizer will provide the sponsor presentation, followed by FDA presentation by Dr. Susan Wallersham on the FDA review of effectiveness and safety of the Pfizer COVID-19 vaccine in infants and children 6 months through 4 years of age. After the lunch break, there will be 1 hour open public hearing, followed by additional question and answer for FDA and sponsor presenters, and committee discussion and voting on Moderna COVID-19 vaccine. And after the break, additional question and answer for FDA and sponsor presenters and committee discussion and voting on FDA on Pfizer-BioNTech COVID-19 vaccine. After that, the meeting will be adjourned. Next slide is this is a question to the committee regarding the Moderna COVID-19 vaccine. Based on the totality of scientific evidence available, do the benefits of the Moderna COVID-19 vaccine when administered as a 2-dose series, 25 microgram each dose, outweigh its risk for use in children 6 months through 5 years of age? Please vote yes or no. This is the question to the committee regarding the Pfizer-BioNTech COVID-19 vaccine. Based on the totality of the scientific evidence available, do the benefits of the Pfizer-BioNTech COVID-19 vaccine when administered as a 3-dose series, 3 microgram each dose, outweigh its risk for use in infants and children 6 months through 4 years of age? Please vote yes or no. Thank you.
Thank you both for your description of our activities today. Next, we go to the Moderna presentation and the FDA presentation concerning, excuse me, we have questions and answers. I just realized a few minutes of questions and answers about the substance of our discussions today. I mean the process, not the substance. few minutes to talk about the process before we get into what I was thinking about going to. So questions and answers about the process. Again, this is a little unusual because we are switching from one product to the other. The reason behind this is that the oral public hearing has to be held at the time it is being held. So we're going to be doing this switch from one product to the other and going back. So we're going to have to pay attention to what we are going to be discussing. Any questions about the process? Right now, I'm looking to see if I have any hands raised. And I do not. So I was right in going and jumping ahead to the presentations. Let's do that now. I'd like to call on Dr. Vinals again, who spoke to us yesterday about the other approach to use of the Madeira vaccine, and she's going to lead, as she did yesterday, her team. Dr. Vinals.
Good morning. My name is Carla Vinals, and I'm the Vice President of Regulatory Affairs Strategy for Infectious Diseases at Moderna. Thank you again to the FDA and VRBPAC for the opportunity to present today our safety, immunogenicity, and efficacy data for mRNA-1273, the Moderna COVID-19 vaccine. We're here today requesting emergency use authorization of mRNA-1273 as a 2-dose primary series for the prevention of COVID-19 in young children, 2 to 5 years of age, and infants and toddlers, 6 to 23 months of age. The proposed 2-dose 25 microgram primary series is to be administered 1 month apart. The totality of safety, immunogenicity, and efficacy data from our clinical development program supports that the benefits of mRNA-1273 in young children outweigh the known and potential risks. mRNA-1273 was generally well tolerated, and the safety profile is consistent with that observed in older adult age groups. No new safety concerns have been identified. Our pediatric studies were designed to meet FDA recommendations for emergency use authorization to infer vaccine effectiveness based on immunogenicity compared to young adults, as the efficacy in adults has already been demonstrated. In both age groups, the co-primary immunogenicity objectives were met. In addition, there was evidence of efficacy against COVID-19 conferred by mRNA-1273 in both age groups, and the rates were comparable to the effectiveness observed in adults during the Omicron period. Our clinical trials enrolled more than 6,600 participants across the 2 age groups, and more than 5,000 have received at least 1 dose of mRNA-1273. The median duration of follow-up in each study cohort is greater than 2 months, which again meets the requirements outlined in the guidance. The dose selected met all immunogenicity objectives compared to young adults, and vaccine efficacy is consistent with what was observed with adults. We have also established plans for extensive follow-up post-authorization to ensure that the long-term safety and effectiveness of mRNA-1273 is closely monitored. Based on this information, we will demonstrate today that the benefits of mRNA-1273 in infants, toddlers, and young children outweigh the potential risks. Here's now the agenda for the rest of our presentation. And I'll now turn the presentation over to Dr. Anderson, who will review the unmet medical need for COVID-19 vaccines in young children.
Thank you, and good morning. My name is Dr. Evan Anderson. I'm a professor of pediatrics and medicine and a practicing physician at Emory University and Children's Healthcare of Atlanta. I'm grateful for the opportunity to present today the burden of COVID-19 in infants and young children and the need for vaccines. This slide lists my conflicts of interest, which have not changed since yesterday. I've been intricately involved with the clinical trials of COVID-19 vaccines, including the Moderna and Pfizer vaccines. As a father of 4, I have a vested personal interest in seeing children protected against COVID. Early in the pandemic, there were several common misperceptions about COVID-19 in infants and young children and its associated risks, as well as the potential need for vaccination. Today, I will review the data that demonstrate that these were clearly misperceptions. First, infants and young children do, in fact, get infected with SARS-CoV-2. This slide shows the incidence of SARS-CoV-2 infections per 100,000 population over time, as reported by CDC. Infants and young children are represented with the yellow solid line. Adults and seniors are shown with the gray dashed and solid lines, respectively. While very few diagnosed cases of SARS-CoV-2 infection were observed early in the pandemic, beginning with the Delta wave and now during the Omicron wave, we have seen a substantial increase in the number of infections among these children. Next, we also know that these infants and young children do get hospitalized with COVID. Again, looking at CDC data, we see a substantial increase in the number of hospitalizations during the Omicron surge among infants and young children less than 5 years of age. Recent data have also shown that there is a substantial burden of hospitalizations in young children. This slide shows the outcomes of COVID-related hospitalizations. Roughly 1 in 4 infants and young children hospitalized with COVID require ICU admission. In addition, while we often hear that hospitalizations among healthy children are uncommon, data demonstrate that over 60% of children 0 to 4 years of age hospitalized with COVID have no underlying medical conditions. Unfortunately, infants and young children can and do die with COVID. As of June 2nd, more than 440 infants and young children aged 0 through 4 have died with COVID as documented by CDC. This is a tremendous burden of disease. 74 deaths in 2020, more than 200 in 2021, and almost 150 in the first 5 months of 2022. What is even more striking is when we place the number of deaths due to COVID into perspective. If we think back to the pre-vaccine era, for vaccines that we are now routinely using, such as rotavirus, hepatitis A, rubella, and varicella, the number of deaths that were occurring in children with these pathogens before implementation of routine vaccination were all less than 60 per year. Flu in the current era ranges up to about 87 deaths per year in children less than 5 years of age. For COVID, since the beginning of the pandemic, we've seen 74 to 221 deaths per year among infants and young children 0 to 4 years of age. As Dr. Marks has already highlighted, what we saw with COVID last year alone was more than double the deaths associated with the 2009 H1N1 influenza pandemic. This is a tremendous burden. Having cared for many children that have been in the ICU on ventilators for COVID and with MIS-C, and having cared for several children that have died of COVID, we need to be able to prevent COVID-19. Finally, although the focus is most often on the morbidity and mortality associated with infection, COVID has dramatically impacted children in many other ways. Masking and social distancing of young children is difficult. Almost 60% of children 0 to 5 years of age not enrolled in kindergarten are routinely cared for, in part, by individuals other than their parents, such as other family members or by daycare. Frequent unexpected disruptions in childcare and schooling have significantly contributed to the daily burden for these families during COVID. COVID crisis-related family hardships have adversely impacted the well-being of our children. Finally, concerns have arisen regarding the neurodevelopment of children born during the pandemic, and increases have been observed in child abuse and mistreatment. So in summary, infants and young children do get infected with SARS-CoV-2. I'd also like to highlight that these infants and children are a continuously renewing population. We now have 2-year-olds that were born after the onset of the pandemic. Over 3.5 million infants are born each year to the in the U.S. And by 6 months of age, these infants are all fully susceptible to COVID. Infants and young children do get hospitalized with COVID, and the surge in hospitalizations with the emergence of the Omicron variant was prominent in our youngest children who have no access to a COVID vaccine. Unfortunately, data also show that infants and young children do get hospitalized with COVID. Approximately 1 in 4 of these will require ICU-level care. Infants and young children do, on occasion, die with COVID. In fact, we have seen 442 deaths with COVID since the start of the pandemic in this age range. These deaths far exceed that for many other pathogens for which vaccines are now available and recommended. Finally, these children and their families have been profoundly impacted in many other ways by COVID. All of this taken together is why a safe and effective vaccine for COVID-19 is needed specifically for our infants and young children. Thank you very much for the opportunity to present to you today. I will turn the presentation over to Dr. Das.
Good morning. My name is Rita Das, and I'm the Vice President of COVID-19 vaccines at Madeira. I'm pleased to present the safety, immunogenicity, and efficacy data from Study 204 in young children 6 months through 5 years of age. Our development program includes more than 5,000 young children who received at least one 25 microgram dose of mRNA 1273. Overall, this represents a substantial pre-licensure safety database in these age groups. Study 204 was conducted in 2 parts. Part 1 was the open-label dose escalation study, and it was conducted to select a dose level for further testing in Part 2, which was the placebo-controlled portion of the trial. Both 25 and 50 micrograms were studied in 2 to 5-year-old children. The 25 microgram dose was chosen for the older children, so the 50 microgram then was not investigated in the younger children. The 25 microgram dose was chosen for both age groups because it showed an acceptable tolerability profile and demonstrated a high likelihood of meeting the pre-specified immunogenicity success criteria. After Part 1 was completed, a DSMB meeting occurred to ensure the committee's concurrence with the selected dose. Part 2 randomized children in a 3-to-1 ratio to receive either mRNA 1273 or saline placebo. The children will be boosted and followed for an additional 12 months. The data we will present today focus on Part 2, which evaluated the 2-dose 25 microgram primary series against placebo. The median safety follow-up meets the EUA recommendations of at least 2 months after the final dose. The Part 1 cohorts had 7 to 8 months of follow-up, and the Part 2 cohorts had median safety follow-up of 2 months post-dose 2. Safety endpoints included solicited local and systemic adverse reactions, which were collected 7 days post-vaccination. All unsolicited events were captured for 28 days after each vaccination, and SAEs, medically attended AEs, and adverse events of special interest were followed throughout the entire study. Vaccine effectiveness was a primary objective, and it was successfully inferred by meeting the predefined immunogenicity criteria, which were agreed with the FDA. There were 2 non-inferiority criteria. First, the lower bound of the GMC ratio had to be at least 0.67, and the point estimate had to be at least 0.8. The FDA requested that if we selected doses lower than 100 micrograms, we ensure that the point estimate of the GMC ratio be at least 1.0. Second, the lower bound of the difference in seroresponse rates, which were defined as a 4-fold rise from baseline titers, had to be greater than -10%, with a point estimate greater than -5%. Evaluation of efficacy was pre-specified as a secondary objective. As in the 301 study, there were two case definitions applied. The CDC definition, which requires one systemic or respiratory symptom, and the 301 definition, which requires two systemic or a single respiratory symptom. Both case definitions require a nasal swab positive by RT-PCR for SARS-CoV-2. The CDC case definition was considered primary since children tend to have less severe symptoms of COVID than adults. Turning to results. Overall, the demographics were well balanced between vaccine and placebo in both age groups. The mean age in the youngest group was about 11 months, and it was 3 years in the older group. Gender, race, and ethnicity also were well balanced. Next, I'll review the safety findings. Starting with the solicited local reactions in children 2 to 5. In this figure, mRNA 1273 is shown in blue and placebo is shown in gray. Pain was the most common event, with similar rates and severity following dose 1 and dose 2. Most local AEs, including pain, were grade 1 to grade 2, with few grade 3 reactions. The median duration of local adverse reactions for this age group was 2 to 3 days. Looking at infants and toddlers, pain was again the most common local adverse reaction, although reports of pain in this youngest group were very similar to placebo and much more similar than the older age groups. Next, turning to systemic reactions. Systemic adverse reactions were evaluated according to age. Young children's events included fever, headache, fatigue, myalgia, arthralgia, nausea, vomiting, and chills. For infants and toddlers, events included fever, irritability and crying, sleepiness, and loss of appetite. Headache and fatigue were the most common systemic adverse reactions among children 37 months to 5 years. Among vaccine recipients, systemic adverse reactions were more frequent post-dose 2 compared to post-dose 1, although this difference now was less pronounced than in the older age groups. Duration was 2 to 3 days, very consistent with the older age groups. In this slide, we are showing the systemic adverse reactions collected for infants and toddlers. On the top are the toddlers aged 24 to 36 months, and on the bottom are the infants and toddlers aged 6 to 23 months. Here, reporting rates of systemic adverse reactions were similar between dose 1 and dose 2. Also, these systemic events were reported at similar rates among vaccine and placebo recipients. I will discuss fever separately since this is particularly important in the assessment of pediatric vaccines. This slide shows fever by increment among children 6 months through 5 years. Overall, fever after any dose occurred in about a quarter of the children. The distribution of temperatures was similar between the two age groups. Reports of fever greater than 40 degrees Celsius were rare. And over the next few slides, I will provide a detailed assessment of all the fevers. In this slide, we have maximum temperatures post-dose 1 and post-dose 2 according to temperature ranges. First, we look at the fevers in children 2 to 5 years. We see that fevers occurred more frequently following the second dose. It's important to note that most reports of fever were less than 39 degrees Celsius. Here are the fevers for infants and toddlers. Again, fevers are reported more commonly post-dose 2. The rates of fever greater than 39 degrees in this youngest age group are very similar to placebo. This figure shows fever by day after the second dose in children 2 to 5 years of age. Most events of fever occurred within 2 days following vaccination. Beyond day 2, we see that fever rates in the children receiving mRNA 1273 are similar to placebo. The median duration of fever in this age group was 1 day. It's important to note that this study was conducted during the winter months when respiratory infections are prevalent, and this is evidenced by the relatively higher rates of fever in the placebo group as well. We see a similar pattern in infants and toddlers, with the peak of fever again occurring on days 1 and 2 after vaccination. On subsequent days, fevers look similar among the vaccine and placebo groups. The higher background rate of fever is even more prominent in the infants and toddlers. There were 15 children with fever greater than 40 degrees Celsius in the mRNA group and 3 in the placebo group. The peak temperature of greater than 40 degrees Celsius had a duration of less than 1 day. Of the 15 events in vaccine recipients, 6 of the children also had symptoms of concurrent viral infections. Since febrile seizures can occur in up to 5% of young children, next I will talk about the febrile seizures that were reported in children 6 months to 5 years. There were 4 episodes of febrile seizures overall in Study 204. 1 was proximal to vaccination and considered related by the investigator. This child also had a maculopapular rash onset 2 days after the seizure and then went on to have a subsequent seizure associated with another fever approximately 6 weeks later. The child has remained in the study and received dose 2 of the vaccine without event. The other 3 events occurred 10 to 66 days after vaccination and were not considered related by the investigators. All 3 events occurred in children with other symptoms of either concurrent viral infections or one child who had a periodic fever syndrome. Next, I will discuss unsolicited adverse events. Presented here are the events reported up to 28 days after any injection in children 2 to 5 years. The incidence of unsolicited AEs was similar among vaccine and placebo recipients. There were no SAEs considered to be related by the investigator. Incidence of MAAEs were similar, and there were no AEs which led to discontinuation of the vaccine or from the study. After the data cut, there was 1 event of urticaria reported on day 1 post-vaccination that did lead to discontinuation. There were no deaths or adverse events of MIS-C or myocarditis. Among the infants and toddlers, the incidence of unsolicited AEs overall and MAAEs were again similar among vaccine and placebo recipients. There was 1 SAE within 28 days, which was considered related to vaccination, which I already described in the fever discussion. Since the data cut of our submissions in late February, we pulled the SAEs from our live database in early May to provide further reassurance of mRNA-1273 safety. This updated analysis did not identify any new safety signals, and there were no SAEs which were considered by the investigator to be related to vaccination. Next, we'll turn our attention to the immunogenicity data. The 2 co-primary immunogenicity objectives were met for children 2 to 5 years old after the 2-dose primary series. The ratio compared to young adults was 1.01 with a lower bound of 0.88. Seroresponse rates were close to 100% in both groups, with a difference of minus 0.4% and a lower bound of minus 2.7%. Next, looking at infants and toddlers 6 to 23 months of age, we see again that both co-primary immunogenicity endpoints were met after receiving the 2-dose primary series of mRNA-1273. The ratio compared to young adults was 1.28, with a lower bound of 1.12. In addition, the seroresponse rate was 100%, a group difference of 0.7% and a lower bound of minus 1.0% was observed. Next, I'll review the efficacy assessment, which was a secondary objective. I'd like to highlight that our pediatric studies were conducted and efficacy follow-up was performed throughout a time when the predominant SARS-CoV-2 strains were changing. And this is important context when interpreting the efficacy results in the 2 youngest cohorts. As shown in this slide, the enrollment and efficacy follow-up in young children was conducted during the Omicron variant wave. The impact of the Omicron daily incidence is apparent from the curve shown in red. From December 2021 through March 2022, the daily U.S. incidence of SARS-CoV-2 infections rose from fewer than 200,000 per day to a peak of 1.4 million cases per day, indicating that the SARS-CoV-2 epidemiology changed significantly when these youngest cohorts were followed. Moving now to the efficacy results. We took a comprehensive approach to capturing cases. All children with symptoms were requested to come into the clinic for illness visits where a nasal swab was collected for RT-PCR. Efficacy estimates in the children 2 to 5 years were based on 180 cases captured over 71 days post-dose 2. There was a lower incidence of COVID-19 by both case definitions in children who received vaccine compared to those who received placebo. Statistically significant efficacy of 36.8% was observed using the CDC definition. When we get to the 301 case definition, we see that the number of cases is reduced and the efficacy is 46.4%. Next, looking at efficacy among infants and toddlers 6 to 23 months. Efficacy estimates here are made using 85 cases captured over 71 days post-dose 2. We again see statistically significant efficacy of 50.6% when using the CDC definition. When we get to the 301 definition, the point estimate is directionally similar, but the confidence intervals are wider due to the drop in the number of cases. To that end, at the start of the Omicron wave, we noticed that parents were reluctant to bring the youngest children into the site for illness visits. Instead, they were calling in results of positive home antigen tests. Since we captured results from the home antigen test as well, we did a sensitivity analysis defining COVID-19 by either positive PCR or home test. With the increased number of cases captured, the confidence intervals narrowed, and the point estimates for efficacy were now 53.5% and 43.7%, with confidence intervals excluding 0. For context, we will now look at real-world effectiveness in adults during the Omicron surge to help interpret the vaccine efficacy from our pediatric program. Presented on the slide on the left are real-world effectiveness data against Omicron among adults. These data are from our collaboration study with the Kaiser Permanente Health System. Vaccine effectiveness of mRNA-1273 against infection was 44% when the Omicron variant was predominant. Now on the right are the estimates of efficacy from Study 204 in infants and young children. Efficacy of mRNA-1273 was consistent with the effectiveness seen in adults. While vaccine effectiveness against any infection was lower during Omicron, we continued to see the benefits of mRNA-1273 against hospitalization. 2 doses of mRNA-1273 was shown to be 84% effective against hospitalization during the Omicron period. This is important because we would expect the same level of protection in children given the consistency of the immune response and efficacy with adults. Study 204 is ongoing, and safety follow-up will continue for all participants. Children will be offered a booster at least 4 months after the second dose. They will be boosted either with mRNA-1273 or our bivalent Omicron-containing vaccine. In summary, mRNA-1273 was well tolerated. Local and systemic reactions were seen less frequently in these youngest groups. Solicited adverse reactions were mostly grade 1 to 2 and slightly more common after dose 2. Fever was most commonly reported in the first 2 days after vaccination and resolved in 1 day. No deaths, myocarditis, pericarditis, or MIS-C were reported among vaccine recipients. There was 1 related SAE of fever, febrile seizure within 28 days of any vaccination. The primary immunogenicity objectives were met. 2 doses of mRNA-1273 were shown to be immunogenic. GMCs and seroresponse rates were non-inferior to young adults. Vaccine efficacy can therefore be successfully inferred based on immunogenicity. In both age groups, direct efficacy against COVID-19 was observed during the Omicron period, again consistent with the effectiveness observed in adults. And now I'll turn the presentation over to Dr. Miller to summarize.
Thank you, Dr. Joss. Good morning to the committee members. My name is Jacqueline Miller, and I'm the Senior Vice President and Therapeutic Area Head for Infectious Diseases at Madeira. And I'd like to summarize our presentation for children 6 months to 5 years of age. Dr. Anderson reviewed a significant unmet medical need remains for pediatric vaccines against SARS-CoV-2, and hospitalizations due to COVID-19 disease have increased amongst the youngest age cohort during the Omicron period. Of these children, approximately 1 in 4 will be admitted to the ICU. Since the beginning of the pandemic, 442 deaths involving SARS-CoV-2 have been reported in children up to 4 years of age, and this exceeds the number of deaths due to other vaccine-preventable diseases in their respective pre-vaccine eras. Vaccine effectiveness has been demonstrated in children 6 months through 5 years of age via immunobridging to the young adult cohort from the 301 study, which demonstrated vaccine efficacy against any and severe COVID-19 disease. The immune response has been remarkably consistent across age groups in a 2-dose primary series with lower doses administered to younger children. This slide depicts the immune responses ranging from young adults to children in all age cohorts, and across the pediatric age groups, the ratio after the second dose ranged from 1.01 through 1.28, successfully meeting all primary immunogenicity hypotheses. Additional support for this EUA submission was provided through the secondary assessments of efficacy, which were comparable with effectiveness in adults for the same variant of concern. Although we did not observe severe cases of COVID-19 in children at the time of the data cutoff, this consistency leads us to believe that efficacy against severe protection or severe disease will be similar to adults, and this will be evaluated in our ongoing post-authorization study. We plan to administer booster doses with our Omicron-containing bivalent vaccine to the 6-month to 5-year-old cohort, which will generate the safety and effectiveness data. Children will be followed for 12 months after boosting. The ongoing post-authorization studies we described yesterday will also be extended to the youngest age cohorts. So in summary, our pediatric development program meets all FDA recommendations for EUA in children 6 months to 5 years of age. Our clinical trials enrolled more than 6,600 participants across these 2 age groups, and more than 5,000 participants have received mRNA-1273 with more than 2 months of follow-up. The 25 microgram dose has met all pre-specified immunogenicity objectives, and vaccine efficacy is consistent with what was observed with adults during the Omicron period, allowing the initiation of protection in infants and young children as of 6 weeks after initiating the vaccination schedule. Our long-term safety and effectiveness studies will continue to evaluate the impact of mRNA-1273 in infants, toddlers, and young children. Based on this information, we have demonstrated that the benefit-risk profile of mRNA-1273 is strongly favorable in children 6 months to 5 years of age. And so we are requesting emergency use authorization of a 25 microgram 2-dose primary series in children 6 months to 5 years of age. This proposal is the result of careful dose selection and the optimization of the immunogenicity and reactogenicity profile in this age group. And the proposed dosing schedule is consistent with our approved dosing schedule in adults. We have heard the feedback from the committee yesterday and want to assure you that all children enrolled in these studies are being boosted and followed for safety, immunogenicity, and disease incidence for 12 months afterwards. And of these, a cohort will contain our Omicron-containing booster, since the data over time indicates that Omicron represents a step change in the evolution of this virus. These booster data will roll out over the summer, and we will be submitting them for FDA review as soon as possible. However, as children under 4 have had the greatest increase in their risk of hospitalization due to COVID-19 during the Omicron surge, initiating this vaccination series now is vital to start protecting children this summer. Thank you very much to the FDA and this committee, as well as all of our collaborators, and particularly to the children and parents in our summer. We heard from one of those parents yesterday, and her story was quite compelling. I'd be happy to take any questions from you now.
Thank you to the Moderna team. Very clear presentations. I'd like to ask the committee to come up with some questions on the specifics. We're going to have a much broader discussion this afternoon after we hear the FDA presentation. So this should be mainly for clarification of questions in the short time we have available right now. Dr. Gans.
Thank you very much for that presentation. I appreciate also your doing information about the boosters. I did have a question just to clarify on maternal antibodies. Are you collecting, at least for the youngest babies at that 6-month mark? I noticed that in some of the immunogenicity, at least from the slides that I looked at, there were several infants that had pre-existing antibodies. And so I'm wondering about, are you collecting information on the mother's immunization status during pregnancy? And are you looking at I'm imagining a pre-vaccine antibody, so that's how you're getting the group that had pre-existing. And are we looking for the distinction between infection and maternal antibodies in those? So the maternal antibody question continues. Are those children that are seeing breakthrough disease, or are there any differences in that group moving forward?
No, thank you for that question, Dr. Gans. So this particular study did not collect maternal antibodies. However, we're initiating a study in infants that are at birth to 6 months of age. And that study called Baby Cove is initiating now. And our intent is to stratify our results by immunogenicity. Or by maternal antibodies.
Okay. In this group, you didn't collect that data, even if a mother got immunized or not.
Yeah.
Thank you.
Thank you. Dr. Portnoy, followed by Dr. Chatterjee. Dr. Portnoy.
Great, thank you. Actually, I learned the trick that you hit the raise your hand early, and that way you can get your question in advance.
That's right.
Exactly. I was just wondering about infants and children who, in your study, who had been previously infected with COVID, and whether there was any effect of a previous COVID infection on immunogenicity and effectiveness of the vaccine. How many of the patients in your study were previously infected and already had some immunity going into the trial?
Yeah, so we actually do have information on those that were previously infected, and previously infected is defined as having either a positive RT-PCR swab or a nucleocapsid protein antigen pre-vaccination. And can you please put up the slide first for children 2 to 5 years of age? It's IM4. But the impacts that we saw in both children 2 to 4 and infants and toddlers 6 to 23 months of age, we did see increases in antibody titers, and in fact, evidence of previous infection actually led to substantially higher antibody titers, and this is really consistent with data that other authors have published, suggesting that a combination of a previous Omicron infection and vaccination actually leads to the longest protection against further Omicron infections.
And were there any differences in adverse events from the vaccine in those who had previously been infected?
The difference was primarily and I'm sorry, can you also put up the infants and toddlers, please? It's IM5 slide, please. Thank you. Just to show you those while I talk through the safety data. We saw a similar reactogenicity profile, but the timing of when those reactions happen is different. So the reactions tended to happen more commonly at the higher rate post-dose 1 versus post-dose 2.
Great, thank you.
Thank you, Dr. Chatterjee, followed by Dr. Cohen.
Yes, thank you. So I have 2 questions, if I may, Dr. Monto. The first one is related to slide 39, I believe. There was mention made of higher fever noted in some of the participants that had symptoms related to other viral infections, potentially other viral infections. The question is, were these participants tested for other viral infections, and do you have those data?
Yeah, so if the children came into the office, the physician may have chosen to test for other viral infections. Because we were obtaining nasal swabs and not E swabs, we don't have the biofire results in this younger population as we do in the older population. But this just represents the concurrent symptoms because the parents report to the physician all of the AEs that are occurring simultaneously. We can say that there were multiple symptoms in those 6 participants.
Okay. My second question is with regard to concomitant administration of other vaccines, particularly for the 6-month-old participants. Were they recipients of other vaccines simultaneously, or were those given at a different time?
Yeah, so the administration of other vaccines was actually given at a separate time. And, you know, the reason for that was when we started this whole endeavor, there were actually a number of questions about the appropriate dose, the use of the mRNA platform, and we really felt that it was important to first select the right dose and tease apart and fully describe that reactogenicity profile. So in the study that we're about to conduct in infants, we are going to be looking initially in infants for the right dose without concomitant vaccination. And then the intent is for those subjects, because obviously as COVID continues with us, the renewable cohort is the birth cohort. We're also going to test versus non-concomitant administration.
One last question, if I may, Dr. Manto. This is follow-up to the answer.
A very quick one.
Yeah, and that is in the children who received other vaccines, were those vaccines given before or after the COVID vaccine?
I think it depended on the physician's choice and how they wanted to administer the schedule. What we asked was that they separate the vaccinations by at least 2 weeks for influenza and a month for the other vaccinations.
Thank you.
Thank you. Dr. Cohen, followed by Dr. Levy.
Thanks, Dr. Miller, for such a clear presentation throughout. I have a question about case ascertainment, and I was wondering if you had any data on the % positives amongst the cases, the vaccine recipients and placebos, and if there was a difference in the number of parents who are bringing their kids in for testing between the 2 groups, and if there was any, and how often parents were bringing kids in for testing versus how often they were positive for COVID.
Yeah, so I should emphasize that parents were really encouraged by the staff to come in, and I think that was incredibly important to the investigators in the study. They did an amazing job, I think, at a difficult time. So we did not analyze data based on whether they came in for or did a home test versus an RT-PCR. What I can show you is the efficacy regardless of symptoms that were reported. So that at least gets at some of the milder versus more serious symptoms. And so first, I'd like to share this slide. It's EF45. Could you put the slide up, please? All right, so these are the results in the 2 to 5-year-olds. And as you can see, the case split is 120 versus 283, and there was a 3-to-1 randomization rate with vaccine effectiveness of 26.9% and a lower limit above 0. And I'm going to have to check on the data for the other age group, and I'll bring that after the break.
Thank you, Dr. Levy, followed by Dr. Bernstein. And unfortunately, at that point, we're going to have to cut off these questions. You'll have a chance later. Dr. Levy.
Yes, thank you for the presentation. If I understood correctly, there were 4 cases of febrile seizures, only one of which was attributed by the investigator as possibly related to the vaccine. For that one case, can you please let us know more about the current status of that infant and how it played out?
Yeah, so this infant was a 17-month-old female. She experienced her seizure 2 days after the first dose. Her maximum temperature was 103.1, and she was noted after that initial fever a day later to have a macular papular rash on covering her body. Her temperature actually reached a Tmax of 104 on day 2. So the seizure happened with 103 temperature approximately 6 hours after her vaccination. She was treated with ibuprofen and paracetamol, was observed in the ER, and then discharged to home. She did actually end up having a second febrile seizure, so that happened about 6 weeks later with other symptoms of fever, respiratory infection. And then she actually did go on to stay in the study, receive the second dose without subsequent seizure. And so I think that's, I mean, she's continued in the study throughout. She's not a discontinuation.
And the rash would be an unusual finding after the vaccine, or you view that as potentially a different?
I think it's hard to say, right? Fever and rash occurs with the vaccine. Fever and rash also occurs with viral syndrome. So certainly something was happening. But she was noted to have an O2 stat of 97%. She was irritable once she got to the ER, but otherwise was okay. And at the time of the seizure, she was noted to be limp with no purposeful movements, but the seizure was not observed by medical professionals.
Thank you.
Thank you. Dr. Bernstein.
Hi, thank you very much to you, Dr. Miller, and to your colleagues for very clear presentations. My question relates to the future studies that you're doing and your discussion about whether these are being termed as boosters or whether this will be a primary series. You're talking about a third dose, and I think it gets a little bit confusing to the public and to others about what a primary series is versus a primary series and a so-called booster. And I was wondering how you're determining that or why you're labeling it as a booster.
Yeah, thank you for that question, Dr. Bernstein. I know this was a topic that came up yesterday. And so I would really like to show the RCC curves from our study in order to maybe discuss that further. But while the team pulls up those RCC curves, so it's slide FF4 and FF5, please. I think the terminology, you can call it a primary series, you can call it a booster dose. I think all of us agree that these children are going to need a third dose at some moment in time. But I think the point I would like to make is by administering these 2 doses on the schedule that we've shown, you begin to see separation of the RCC curve between the mRNA 1273 group in red and the placebo group in blue by day 40 in the modified intent to treat cohort. So clearly the 2-dose series is initiating protection early on after the schedule administered at day 0 and day 28. And, certainly appreciate the points that are being made by the committee and are really committed to not confusing the public. But at this moment, our view is it's just critically important to start vaccinating babies so that they can start benefiting from the same protection as other age cohorts. And can I also show FF5, please, just to be complete in the 2 different age cohorts?
Okay.
Thank you.
Thank you. You see it?
Yep. Thank you.
Okay. Did you have anything further to add about the second slide?
No, just to show that again, by 2 weeks after the second dose, we're beginning to see separation in those two RCC curves, which I think ultimately is the objective of initiating immunization in these children.
Thank you, and apologies to those who we haven't had time to include in our questions session. We'll have much more time later after lunch. Now we go to the FDA presentation, the review of effectiveness and safety of Moderna COVID-19 vaccine in infants and children 6 months through 5 years of age. Dr. Wish.
Thank you. Good morning. I'm Robin Wish. I'm a medical officer in the Center for Biologics, Office of Vaccines Research and Review, Division of Vaccines and Related Products Applications at FDA. I will be presenting FDA's review of the effectiveness and safety of the Moderna COVID-19 vaccine in children 6 months through 5 years of age, submitted under an emergency use authorization amendment. I'd like to start off by acknowledging the many contributions of my colleagues in CBER. Here is the outline of my presentation today. I will start with regulatory background and then cover the design of the study submitted to support emergency use authorization for use of the Moderna COVID-19 vaccine as a 2-dose series in children 6 months through 5 years of age. I will review the Part 1 dose selection data and the Part 2 immunogenicity, descriptive efficacy, and safety results. Then I will provide a summary of the planned pharmacovigilance activities and conclude with an overall summary of benefit risk for the 6 months through 5 years age group. We'll start with background. The Moderna COVID-19 vaccine contains nucleoside-modified mRNA that encodes for the full-length spike protein of SARS-CoV-2 encapsulated in lipid particles. It was licensed as Spikevax for individuals 18 years of age and older on January 31st, 2022. Data included in the EUA request for children 6 months through 5 years of age were from study P204, a phase 2-3 study with an initial open-label dose escalation and age de-escalation phase, followed by a randomized, observer-blind, placebo-controlled phase to evaluate the safety, reactogenicity, and effectiveness of mRNA 1273 vaccine in healthy children 6 months through 11 years of age. Today, I will cover the 6 months through 5 years age groups. This pediatric age group blinded follow-up was through the data cutoff of February 21st, 2022. Data included in the EUA request for children 6 months through 5 years of age were from study P204, the phase 2-3 study with an initial open-label dose escalation and age de-escalation phase, followed by a randomized observer-blind placebo-controlled phase to evaluate the safety, reactogenicity, and effectiveness. Excuse me, I'm so sorry. I'm off slides. This slide provides an overview of the pediatric studies in age groups from 6 months through 17 years of age. Yesterday, the age groups from 6 years through 17 years of age were discussed. Today, I will focus on the age groups from 6 months through 5 years of age, including approximately 1,700 vaccine recipients in the 6 through 20 months age group, which I will refer to as the infant toddler group, and 3,000 vaccine recipients in the 2 through 5 years age group, which I will refer to as the preschool group. In Part 1 of Study P204, the open-label dose escalation age de-escalation phase, enrollment of participants 2 through 5 years of age began with a 50 microgram dose level. Based on the observed rate of solicited adverse reactions, in particular the rates of fever after vaccination, the study proceeded to enroll the remaining Part 1 participants into a lower 25 microgram dose level. Based on the high rate of solicited adverse reactions in the preschool group at the 50 microgram dose level, all Part 1 participants in the 6 through 23 months of age group received a 25 microgram dose. The immunogenicity results of the 25 microgram dose level in participants in the infant toddler group, along with the more tolerable reactogenicity profile of this dose level, supported the selection of 25 micrograms as the dose for advancement into Part 2 for both age groups. Part 2 was the randomized placebo-controlled observer-blind evaluation of a selected 25 microgram dose for each age group, with just over 4,000 participants randomized in the preschool group and approximately 2,300 participants randomized in the infant-toddler group. Participants were randomized 3-to-1 to receive 2 doses of 25 micrograms of mRNA 1273 or placebo given 1 month apart. These are the study objectives and endpoints. The safety endpoints included solicited adverse reactions collected for 7 days after each vaccination in an e-diary and collection of unsolicited adverse events for 28 days after each dose. Medically attended adverse events, serious adverse events, and adverse events of special interest were collected for the entire study duration. There was also active monitoring for myocarditis and pericarditis throughout the study as described yesterday. Using an immunobridging approach, GMCs and seroresponse rates 1 month post-dose 2 were compared to young adults 18 through 25 years of age with demonstrated efficacy from study P301. There were also descriptive efficacy endpoints analyzed as secondary endpoints. For the immunobridging analyses, participants in the per-protocol immunogenicity subset were PCR negative and/or seronegative for SARS-CoV-2 at baseline. Immunobridging to the young adult cohort in study P301, in whom vaccine efficacy was demonstrated during a time period when the original strain was predominant, was based on comparisons of neutralizing antibody responses to the ancestral strain, which carries a D614 gene mutation. The first co-primary immunogenicity endpoint was GMC ratio of SARS-CoV-2 neutralizing concentrations in the specified age group, either 6 to 23 months of age or 2 to 5 through 5 years of age, to those in young adults 18 through 25 years of age. The success criteria required a lower limit of the two-sided 95% confidence interval for the GMC ratio of greater or equal to 0.67 and a point estimate of the GMC ratio greater or equal to 0.8. The second co-primary immunogenicity endpoint was difference in seroresponse rates between participants of the specified pediatric age group and the young adult age group, where seroresponse was defined as greater than or equal to a 4-fold rise from baseline. The immunobridging success criteria required a lower limit of the 95% confidence interval for the difference in seroresponse rates for each of the 2 pediatric age groups minus the young adult age group, of greater or equal to negative 10% and a point estimate of difference in seroresponse rates of greater or equal to negative 5%. For your reference, this slide again provides the definitions of CDC-defined COVID-19 and COVID-19 as defined in study P301, the 2 case definitions assessed in the descriptive efficacy analyses in study P204. Also for your reference, as presented yesterday, these are the most pertinent pediatric analysis populations used for evaluations of immunogenicity, efficacy, and safety. This slide provides the follow-up time for study participants calculated from dose 2 to the cutoff date of February 21st, 2022. In the infant toddler group on the top of the slide, the median follow-up time from dose 2 was 68 days. In the preschool group at the bottom of the slide, the median blinded follow-up time from dose 2 was 71 days, and the median follow-up time from dose 2, including both blinded and unblinded follow-up, was 74 days. The demographics and baseline characteristics of participants in the infant toddler group are displayed in this slide. Demographic characteristics were comparable between the vaccine and placebo groups. The majority of study participants were white and non-Hispanic. Most participants in the study were enrolled in the U.S. Approximately 20% of study participants were obese, and approximately 6% had evidence of prior SARS-CoV-2 infection at baseline. The demographics and baseline characteristics of participants in the preschool group are displayed here. Similar to the previous slide, demographic characteristics were comparable between the vaccine and placebo groups. The majority of study participants were white and non-Hispanic, and most participants in the study were enrolled in the U.S. Approximately 11% of study participants were obese, and approximately 9% had evidence of prior SARS-CoV-2 infection at baseline. I'll now move on to discussing immunogenicity data. Shown here is the co-primary endpoint of the ratio of neutralizing antibody GMCs in the infant toddler group compared to young adults at 4 weeks post-dose 2. The study met the pre-specified success criteria of lower bound for GMC ratio greater or equal to 0.67 and point estimate greater or equal to 0.8, with a lower bound of 1.1 and a point estimate of 1.3. This slide shows the co-primary endpoint of difference in seroresponse rate in the infant-toddler group compared to young adults. The study met the pre-specified success criteria of lower bound greater or equal to negative 10% and a point estimate greater or equal to negative 5%, with a lower bound of negative 1 and a point estimate of 0.7. The GMC ratio and difference in seroresponse rates across demographic subgroups were consistent with the results obtained based on the general study population, though some of these analyses were limited by small subgroup size. Results for subgroup analyses of the GMCs in the infant-toddler group by baseline SARS-CoV-2 status are displayed here. The small number of participants with positive baseline SARS-CoV-2 status in the immunogenicity subset had numerically higher GMCs at day 57 compared to those negative at baseline, consistent with the immunogenicity results observed in the 18 through 25 years age group. Now we move to the preschool group. Shown here is the co-primary endpoint of a ratio of neutralizing antibody GMCs in the preschool group compared to the young adults at 4 weeks post-dose 2. The study met the pre-specified success criteria of lower bound for GMC ratio greater or equal to 0.67 and point estimate of GMC ratio greater or equal to 0.8, with a lower bound of 0.9 and a point estimate of 1. This slide shows the co-primary endpoint of difference in seroresponse rate in the preschool group compared to young adults. The study met the pre-specified success criteria of lower bound greater or equal to negative 10% and a point estimate greater or equal to negative 5%, with a lower bound of negative 2.7 and a point estimate of negative 0.4. The GMC ratio and difference in seroresponse rates across demographic subgroups in this age group were also generally consistent with the results obtained based on the general study population, though some of these analyses were also limited by small subgroup size. Results for subgroup analyses on the GMCs and the children in the preschool group by baseline SARS-CoV-2 status are displayed here. Again, participants with positive baseline SARS-CoV-2 status had numerically higher GMCs at day 57 compared to those negative at baseline, consistent with immunogenicity results observed in 18 to 25 years of age. I'll now move on to the descriptive efficacy data. Vaccine efficacy was descriptively analyzed as a secondary endpoint in the study with the data cutoff of February 21st, 2022, and during a period when the Omicron variant was the predominant circulating strain in the U.S. Shown here are vaccine efficacy results for first occurrence COVID-19 starting 14 days after dose 2 based on the CDC and P301 case definitions. No severe COVID-19 cases were reported during the study in the infant-toddler group. Among the approximately 6% of total study participants with evidence of prior SARS-CoV-2 infection at baseline, one placebo participant and no vaccine participants developed COVID-19 starting 14 days after dose 2. Analysis of vaccine efficacy, including a population of participants both with and without evidence of prior SARS-CoV-2 infection or with an unknown baseline status, was similar to the efficacy results displayed on this slide. These are the vaccine efficacy results for the first occurrence of COVID-19 starting 14 days after dose 2 based on the CDC and P301 case definitions for the preschool group. In this group, there were also no severe COVID-19 cases reported during the study. Among the approximately 9% of total study participants with evidence of prior SARS-CoV-2 infection at baseline, one placebo participant and 6 vaccine participants developed COVID-19 starting 14 days after dose 2. Analysis of vaccine efficacy, including a population of participants with and without evidence of prior SARS-CoV-2 or with an unknown baseline status, was similar to the efficacy results displayed here. I will now move on to the safety data. Shown here are the frequencies of solicited local reactions in the infant-toddler group following each dose. Local adverse reactions generally occurred more frequently and were more severe after dose 2 compared to after dose 1, although grade 3 events were uncommon. The most solicited local adverse reaction was injection site pain. Solicited local adverse reactions persisting beyond 7 days after any dose were reported more frequently in the vaccine group than the placebo group, and the majority of events were mild. This table shows the frequency of solicited systemic reactions after each dose in the same age group. The frequencies of systemic reactions were generally comparable across doses, and most events were mild to moderate in severity. The most common solicited systemic adverse reaction reported in the vaccine group was irritability and crying. Grade 4 events were rare and only occurred with the adverse reaction fever. For all solicited reactions in this age group, local and systemic, the majority of events had onset within 1 to 3 days post-vaccination and resolved within 2 to 3 days. Most events that persisted beyond the 7-day reporting period were mild. Overall, the frequencies of solicited reactions were similar among participants with positive and negative baseline SARS-CoV-2 status, except for fever, which was more common in those who were baseline seropositive. Now we turn to solicited adverse reactions in the preschool group. In this age group, solicited local adverse reactions generally occurred more frequently after dose 2 compared to after dose 1. Adverse local reactions tended to be more severe after dose 2, but grade 3 events again were uncommon. The most common solicited local adverse reaction was injection site pain in this age group as well. Solicited local adverse reactions persisting beyond 7 days after any dose were reported more frequently in the vaccine group than in the placebo group. The majority of events were mild. For the 2 to 5 years of age group, the solicited systemic reaction terms differed for participants from 24 through 36 months of age and from 37 months through 5 years of age, as showed in the previous presentation. This slide shows the systemic reactions in the 24 through 36 months of age subcohort. Overall, the frequency of solicited systemic adverse reactions were comparable across doses, with the exception of fever, which was reported more frequently after dose 2. The most common solicited systemic adverse reaction reported in the vaccine group was irritability and crying. Most events were mild to moderate in severity, and grade 4 events were rare and only occurred with the adverse reaction fever. The next 2 slides show the systemic reactions in the 37 months through 5 years of age subcohort. Solicited systemic adverse reactions generally occurred more frequently and were more severe after dose 2 compared to after dose 1, although most events were mild to moderate in severity. The most common solicited systemic adverse reaction reported in the vaccine group was fatigue. And as with the other age groups, grade 4 events were rare and only occurred with the adverse reaction fever. This slide shows the remaining solicited systemic reactions in the 37 months through 5 years of age subcohort. For the entire 2 through 5 years age group, most local and systemic adverse reactions had onset 1 to 2 days post-vaccination and resolved within 2 days after onset. The majority of events that persisted beyond the 7-day reporting period were mild. As in the infant-toddler age group, overall, the frequencies of solicited reactions were similar among participants with positive and negative baseline SARS-CoV-2 status, except for fever, which was more common in those who were baseline seropositive. This table presents the frequencies of unsolicited adverse events in the infant-toddler group. Overall, rates of unsolicited adverse reactions were similar across groups. Unsolicited events reported by at least 1% of participants in the vaccine group and by a higher proportion of the vaccine group compared to the placebo group included injection site reactions and some common childhood illnesses, such as acute otitis media and croup. The most commonly reported unsolicited AEs among vaccine recipients were upper respiratory infection, irritability, fever, and teething. As discussed in yesterday's presentation, symptoms of myocarditis and pericarditis were solicited for the duration of the study through scripted safety calls conducted at 7 days after each dose and every 4 weeks thereafter. This resulted in enhanced reporting frequency of associated symptoms in study P204 compared to those reported in earlier studies in adults and adolescents. The same search strategy as described yesterday was also used for evaluation of the safety data set for participants 6 months through 5 years of age. In the infant-toddler group, neither the captured event of dyspnea nor the events of irritability and vomiting shown on the slide were identified in the additional analyses met the CDC criteria for probable or confirmed myocarditis or pericarditis. While some respiratory tract-related infections were reported with greater frequency in the infant-toddler vaccine group than in the placebo group, analyses including all respiratory tract-related infection preferred terms with and without COVID-19 showed generally comparable rates between the two groups. As shown in the slide, events of croup, RSV, pneumonia, and pneumonia were reported with greater frequency in the vaccine group compared to the placebo group, but there was no pattern concerning time to onset or dose number for these events, and there is not a clear biological mechanism that would explain a causal association for certain respiratory infections but not others. Overall, the frequency and clinical course for these events did not appear unusual given the age group of the study population and the season, fall through winter, during which the study took place. There was also an imbalance in lymphadenopathy-related events in the vaccine group compared to the placebo group, which were reported by 1.5% of vaccine recipients and 0.2% of placebo recipients. This imbalance is consistent with the imbalance observed for solicited events of axillary or groin swelling and tenderness. There were no reported events of anaphylaxis related to study vaccine. This table presents the frequencies of unsolicited adverse events in the preschool group. Overall, rates of unsolicited adverse events were similar across groups. Unsolicited events reported by at least 1% of participants in the preschool vaccine group and by a higher proportion compared to the placebo group included injection site erythema. The most commonly reported unsolicited AEs among vaccine recipients were upper respiratory tract infection, rhinorrhea, and cough. Regarding cardiac events in the preschool group, none of the events captured met CDC criteria for probable or confirmed myocarditis or pericarditis, and no other events were identified in the additional analyses. One participant underwent evaluation by a cardiologist. A 4-year-old male participant with chest pain 5 days after dose 2 that resolved within 30 minutes. That evaluation included a physical exam, EKG, and troponin, which were all reported to be normal. The majority of events in the study were nonspecific in nature, and many were associated with concurrent symptoms, including respiratory tract infections or allergies. In this age group, events of pneumonia and RSV infection were reported with greater frequency in the vaccine group than in the placebo group. Again, there was no pattern concerning time to onset or dose number for these events, and analyses including all respiratory tract-related infection-preferred terms, with and without COVID-19, showed generally comparable rates between the two groups. Overall, the frequency and clinical course for these events did not appear unusual given the age group and the study population and the season during which the study took place. And again, there is not a clear biological mechanism that would explain a causal association for certain respiratory infections but not others. Events of abdominal pain occurred in less than 1% of participants in each group, but were reported more frequently in the vaccine group, 0.7% of participants, compared to the placebo group, 0.4% of participants. The 3 events assessed as related, 2 in the vaccine group and 1 in the placebo group, occurred within 2 days of vaccination, and as discussed yesterday, were likely to be manifestations of systemic reactogenicity. There was also an imbalance in lymphadenopathy-related events, which were reported by 0.9% of vaccine recipients and less than 0.1% of placebo recipients. This imbalance is consistent, again, with the imbalance observed with solicited events of axillary or groin swelling and tenderness. There were no reported events of anaphylaxis related to study vaccine. In the infant-toddler group, there were no reported deaths, and overall, there were few reported serious adverse events, or SAEs, 0.9% in the vaccine group and 0.2% in the placebo group. FDA assessed all SAEs in this age cohort as unrelated, with the exception of the events previously discussed of pyrexia and febrile convulsion that occurred within 2 days of dose 1 in a 1-year-old female participant, followed by the occurrence of a maculopapular rash that was considered possibly related to study vaccine with a plausible alternate etiology of a viral illness. For the 2 to 5 years of age group, there were also no deaths reported. And overall, there were few reported SAEs, 0.3% in the vaccine group and 0.2% in the placebo group. Most SAEs were consistent with events typical in this age group and for the season during which the study took place. FDA agreed with the investigator assessments that none of the reported SAEs were considered related to study vaccine. I'm now going to move on to pharmacovigilance. The sponsor submitted a pharmacovigilance plan to monitor safety concerns that could be associated with the Moderna COVID-19 vaccine. They identified anaphylaxis, myocarditis, and pericarditis as important identified risks, and vaccine-associated enhanced disease, including vaccine-associated enhanced respiratory disease, as important potential risks. Areas the sponsor identified as missing information included use in pregnancy and lactation, vaccine effectiveness, long-term safety, interaction with other vaccines, use in immunocompromised or frail patients or patients with autoimmune inflammatory disorders, and use in pediatric individuals less than 6 months of age. Pharmacovigilance activities under the EUA include adverse event reporting, which may come from vaccine recipients, vaccination providers, the sponsor, or the CDC VSafe program. Both the sponsor and vaccine providers administering Moderna COVID-19 vaccine must report the following information to theirs: serious adverse events irrespective of attribution to vaccination, cases of multisystem inflammatory syndrome, and cases of COVID-19 that result in hospitalization or death. Additionally, the sponsor submits reports of myocarditis and pericarditis as 15 days reports to theirs. The sponsor will also conduct periodic aggregate review of safety data that I will discuss in an upcoming slide and submit periodic safety reports at monthly intervals for FDA review. Furthermore, the sponsor has planned surveillance studies that are summarized on the next slide. There are 4 post-authorization safety studies of myocarditis and pericarditis shown here, including subclinical myocarditis, and 1 post-authorization vaccine effectiveness study that includes individuals 6 months through 17 years of age. I've already presented adverse event reporting under EUA may come from vaccine recipients, vaccination providers, or the sponsor. Reports from vaccine recipients are voluntary, while adverse event reporting by vaccination providers and the sponsor is mandatory. Periodic aggregate safety reports are required to contain a narrative summary and analysis of adverse events submitted during the reporting interval, including interval and cumulative counts by age groups, special populations, and adverse events of special interest. A narrative summary and analysis of vaccine administration errors, whether or not associated with an adverse event that were identified since the last reporting interval, newly identified safety concerns in the interval, and actions taken since the last report due to adverse experiences. Both FDA and CDC will take a collaborative and complementary approach on reviewing adverse events. In the initial stage of post-authorization surveillance, FDA will individually review all serious adverse events on a daily basis. FDA will also examine other sources for adverse events, such as the literature, and will perform data mining to determine if adverse events are disproportionately reported for the candidate vaccine compared to all other vaccines and bears. Any potential safety signals identified will be investigated. And now for a summary of benefits and risks. This slide presents a summary of benefits and risks of the Moderna COVID-19 vaccine when administered as a 2-dose series, 25 micrograms each dose, in children 6 months through 5 years of age. Known and potential benefits include prevention of symptomatic COVID-19 based on immunobridging to young adults, as well as supportive evidence of vaccine efficacy against symptomatic COVID-19, with expected greater effectiveness against more severe disease. Effectiveness against emerging variants and duration of protection are not yet known. Known and potential risks include symptoms of reactogenicity, potential myocarditis, pericarditis, and hypersensitivity reactions. Uncertainties remain regarding adverse reactions that are uncommon or require longer follow-up to be detected. The voting question for today regarding the Moderna COVID-19 vaccine for use in children 6 months through 5 years of age is following. Based on the totality of scientific evidence available, do the benefits of the Moderna COVID-19 vaccine when administered as a 2-dose series, 25 micrograms each dose, outweigh its risks for use in children 6 months through 5 years of age? And that brings me to the end of my presentation. Thank you.
Thank you, Dr. Wish, and you have given us a fair amount of time for you to be questioned because of your succinct presentation. Dr. Levy, is that you raising your hand?
Hi, thanks for the excellent presentation. I had a question about what appeared to be an imbalance with respect to RSV infections and pneumonia. And my question to you is, a priori, we might not think that that's possible. On the other hand, vaccines can have off-target effects on innate memory, so who knows? But just looking at the data, is that a statistically significant higher RSV and pneumonia signal in the vaccine group? And then the lens on the far right, if you could pull up that slide, was a little bit to me because at the same time it said an overall analysis of respiratory infections did not show a difference. So can you talk us through that and let us know what you think?
Sure. Let me go through. I'm going to go through my notes. I don't have the slides to pull up individually for just a second. But so when we did an analysis looking at all respiratory tract infection preferred terms for the entire study, in the 6 to 23-month age group, we saw basically the same number of events in each group. So 21.5% in the vaccine group versus 21.4% in the placebo group. In the 2 to 5 year age group, we saw a similar finding where the rate of all respiratory tract infection preferred terms were similar between the two groups, around 17%. That's for the entire study period. When you looked at individual preferred terms under different types of respiratory tract infections, there were some imbalances. The ones that were pointed out were imbalances where we saw a bit more in the vaccine group compared to the placebo group, but there were also other events that were seen more frequently in the placebo recipients as compared to the vaccine recipients. I think overall our assessment was to think that these events were in a low enough percentage of participants and were common events in this age group and at the season that we did not consider any of these signals that were picked up. I don't know if any of my colleagues or Madeira would like to chime in and add to that.
Just your colleagues right now. Let's not confuse that.
I'd be happy to add a bit to that. So, yes, respiratory infections are also something that we looked into as well with the imbalances that were noted, established. And you had a question about statistically significant. So I will say that because of the large number of comparisons of discrete Madeira terms, these analyses are descriptive. So there's no adjustment made for multiplicity in all of the analyses in the two age groups. The other thing I would point out is that there was an imbalance in the other direction for COVID-19 infections. And that really led to the rates of upper respiratory tract infections actually being evenly distributed between the two groups. So in children who were 2 to 5 years of age, the rate was 9.2%. In the placebo group, 8.1%, in the mRNA 1273 group. And in 6 to 23-month-olds in the placebo group, the rate was 12.2%. The rate with mRNA 1273 was 10.3%.
Dr. Fink.
Thank you. I was going to make some of the same comments about the statistical considerations for these numerous preferred term analyses. But that being said, I do think that if the vaccine were to be authorized for use in this age group, clearly we would want to continue looking at these types of events in post-authorization surveillance. We're not highly concerned about a couple of imbalances in one direction in specific events of infections that are common in this age group. We did not see increased severity of these types of infections in the vaccine group compared to the placebo group. But it is something that I think bears keeping an eye on in the post-authorization safety surveillance. Thank you.
Thank you.
Thank you all, Dr. Meisner, followed by Dr. Marasco.
Thank you, Dr. Montel. And Ofer, I guess, got his hand up before I did, because I had a question similar. That is both slide 37 and slide 40 note that the listed diagnoses are higher in the mRNA 1273 group than they are in the placebo, but then the comments seem to say the opposite. So that's a little bit confusing. But I appreciate your explanation and clarifying that. And the reason that it's important, I think, is because the effect of COVID-19 or the SARS-CoV-2 pandemic on other respiratory viruses such as RSV is kind of interesting. It may the disappearance of the disease may not have all been due to non-pharmacologic interventions. There may have been some viral interactions that reduced other viruses, such as RSV and influenza. So it is an interesting issue as to what's going on here, but thank you for the clarification.
Dr. Marasco, followed by Dr. Chatterjee.
Hi, thank you very much, Dr. Monto, and I'd like to address a couple of questions to the committee, to the FDA, and I'm happy to have Moderna's input into this. So the viral efficacy, the vaccine efficacy against Omicron is lower than one would expect with Wuhan. And this is their first antigenic exposure. And I don't want to be an immunologist aficionado here, but it's pretty clear from influenza data, for example, that the first virus that you get exposed to is going to bias your immune response for the rest of your life. It's called immune imprinting. So when we're vaccinating with an ancestral strain and we're testing against a boosted strain, there's an expected loss of efficacy. And this is because this is really antigenic shift, not drift like we see in the seasonal influenza. This is more like what happened in pandemic 2009. So the real question is, are we gathering data on this? And my concern is, if we just do this blindly, and maybe this is to Moderna, if we're just looking at titers, really are we going to understand the breadth of the repertoire that is being developed and whether we're biasing our protection against one lineage against another? And that's really the essence of my question. It's a matter of, in 2022, the type of data that we're collecting for these studies. This is really more than just serologic to be able to get to the heart of the problem and the heart of what we're doing. Thank you.
Thank you. You've asked a very broad question that needs long-term follow-up in general, not just in terms of this product. Dr. Wish?
Thank you for that.
Sorry, please go ahead. I apologize.
No, absolutely. Go ahead.
I was just going to comment that I think it's a really excellent question, and I think it's one that will be evaluated over time, as Dr. Monto referred to. So I wanted to mention that we will see the effectiveness or efficacy, field efficacy of mRNA 1273 against Omicron in a study that we are conducting actually in partnership with South African Research Council in South Africa. So really where the Omicron variant first emerged, and actually now we're boosting healthcare workers with mRNA 1273 during the BA.4, BA.5 period. And I think those long-term effectiveness studies are really the best way to understand how a vaccine formulation is going to perform against emerging variants of concern. But given that at a certain moment in time, what we're left with is being able to look at immune responses, I'd like to show, if I could, slide BF12, which reflects some of the data that we've been generating with respect to other variants in the youngest age group. So we've been looking actually throughout at neutralizing titers in our older subjects. At the moment, our Omicron neutralization is primarily being performed against the bivalent vaccines that this committee will discuss in 2 weeks. But what we do have are some binding data with respect to the various variants, and I see the computer is thinking about it. There we go. So this is a complicated slide, but what you see in the 4 different rows are the MS, so a multiplex system binding antibody to the 4 key variants. So first starting with the ancestral strain, then the variant, then in blue, the Delta variant, and then in purple, the Omicron variant. And then moving left to right, what you see are the children.
Why don't you wait until we can see it? We're not seeing it yet.
Oh, I apologize. I thought I'm able to see it, so I thought you were as well.
No, we're not.
I have it. But
we can see it,
Some
Arnold.
of us are seeing it, Arnold.
Okay, you can see it? Okay, go ahead.
Yeah, Arnold, some of us can see it.
Thank you.
Okay,
ahead,
my not able. Okay, good.
Dr.
Good.
Wish, just to clarify, this is with the Omicron vaccine. Just to clarify.
No. This is with mRNA 1273. These are samples for studies that you are doing today, and so we have tested children. Or sera from children for all of the different variants, because as you mentioned, while Omicron is our particular consideration today, I think that it's likely that this virus continues to evolve over time. So you see different variants in different colors. The Omicron is in purple at the bottom of the slide. And the dotted line in each of the graphs represents the limit of detection of the assay. And so in all cases, what we're seeing is an increase against the variants of concern. I think the question around what is the right way to prime individuals is a very good one. And it's why we are going to start a primary vaccination study with the bivalent to see if the primary series looks different in terms of antibodies that are generated versus the original mRNA 1273 vaccine.
Thank you. And I assume you have some neutralization assays on some of them, at least.
Yeah,
well.
so
I don't want you to present them. I just you're following up. Okay. Dr. Wish, do you have anything in addition to add?
No, I'd add beyond that. Thank you.
Okay, Dr. Chatterjee, followed by Dr. Kim.
Thank you, Dr. Monto. I have two questions. I have the suspicion that the sponsor might need to weigh in, but the first one is on slide 29 on Dr. Wish's presentation, and this was with regard to the local reactions in the younger cohort. I was wondering how pain was determined in preverbal children. Was that inferred in some way, or how would they know if a 6-month-old had pain?
The parents and caregivers of the children provided e-diaries. I would have to, again, defer to the sponsor to see if they can give a more precise explanation of how parents assess that.
Okay.
I would think it would be, through.
Yeah, so just to make sure I understood the question is how we understand how pain is measured in 6-month-old children? Yes.
Preverbal children. Mm-hmm.
Yeah, because you're absolutely right. Obviously, they can't complain about pain, but it has to do with the parent's assessment of the level of discomfort. For example, when the child's arm or leg is moved, are they crying? So to maybe give you the different grades, grade 1, mild discomfort touch, where maybe there's a reaction when things are touched, moving to grade 2, that they're crying when their limb is moved, and then significant pain at rest, preventing just normally what the child is doing is grade 3.
Okay, thank you.
Dr. Kim, followed by Dr. Hildreth.
Well, thank you very much, Dr. Monto, and that was a terrific presentation, Dr. Wish. I have a question on your slide number 26 and 27, and this, I think this question would also apply to our Madeira colleagues. The efficacy study for the infants and toddlers was 50%, using the CDC definition, and using the study definition from Moderna 301 was 31%. And on the following slide, 27, looking at things for the preschool age group, we're talking 36% and 46% respectively for CDC and Study 301. Now, there's an overlap between these two efficacy results, but it's interesting that the CDC definition and the Study 301 definition basically are inversely related between the infant toddler group and the preschool group. And having looked at the data much more closely, to what, if any, attributes have you seen in the findings that might explain this opposite direction vaccine efficacy findings between these two age groups, and perhaps our Moderna colleagues can add to that.
Other than small numbers and chance, please, Dr. Wish.
Sure. Thank you for that. So, yes, if you look at the two numbers, Dr. Monte is where I'm thinking as well. You can see, the P301 definition is a more stringent definition of COVID-19 disease. And so the numbers are smaller for both age groups for those diagnosed with P301 definition as compared to the CDC definition. If you look at the vaccine efficacy for the younger 6 to 23 months, that the confidence interval is much wider and crosses zero. So, it's hard to interpret the reliability of that vaccine efficacy for that population with that definition. But we did see the trend that of course, there were fewer cases with the P301 definition, and it's difficult to tell because of the small case numbers using that more stringent definition in this population. But I defer to Moderna if they want to add to that. Thank you.
Let's go ahead so we can get to the bottom of our list of questioners. Dr. Hildreth, followed by Dr. Fuller, who will have the last question.
Thank you, Dr. Monto, and thank you, Dr. Wish and Dr. Benals, for your great presentations. My question is related to the just prior question that was asked. The data shows that the efficacy for the toddlers and the infants have much lower efficacy than the 6 to 11-year-olds. But the data you've shown us show that they all have about the same geometric titers for neutralization. So is there a disconnect there that you can help me understand? The neutralizing antibody titers are clearly the same, but the efficacy is not. So I just would like you to help me understand that.
Thank you for that question. The one thing I can say is that in the time period when the infants and toddlers or the younger pediatric population were being evaluated was during Omicron, whereas I believe the 6 to 11-year-old age group was evaluated during the time of the Delta surge. So there is that difference in time period as far as efficacy of the vaccine against the various variants. I don't know if anybody else again.
During the pseudotype virus neutralization assay, did you do a side-by-side comparison of Omicron and Delta to confirm that?
I would have to defer to Moderna for that.
Okay.
A quick response. We're going to have time to go into this in detail later on. So please, quick response, because I'd like to get to Dr. Fuller.
Sure. Great question, and mentioned we're in the process of generating the neutralization data against Omicron and the other variants with respect to the youngest kids. We did review a slide yesterday, BF11 in the older children, just to show you how it looked across age groups versus the ancestral strains. So can I have the slide up, please, BF11?
Okay, let's do it quickly.
Just real quick, I promise.
Okay.
Just to answer to Dr. Hildreth's question, which is, are we looking into Omicron? We continue to look at neutralization titers across age groups and also as new variants emerge.
Okay, thank you.
Thank you.
Thank you. Dr. Fuller, final question before the break.
Yes, so you mentioned that you looked at obesity as an underlying factor. Did you look at things between the 2 and 6-year-olds like sickle cell or asthma that might be underlying conditions and the results of the vaccine in those?
Yes, there were the underlying comorbidities that were looked at, including obesity, were also respiratory, chronic respiratory conditions, including asthma and cardiac conditions. The numbers of children in each of those subgroups were very small, so it was hard to come to conclusions about differences given the very small numbers.
But things like sickle cell or juvenile diabetes that can be diagnosed early, they did not look at.
Yes, I'm sorry, I apologize. Diabetes was included. I don't recall seeing sickle cell disease in those baseline comorbidities. I'm not sure of that. I can pull up. I know in the briefing document there is a table of baseline comorbidities, and then we'll show you that. I can't recall if there were cases of diabetes. They were very, very small. It wouldn't be on these slides. It's in the briefing document. I can find that information and get back to you.
Thank you.
Okay.
Sure.
Thank you all. It's time for the break. And we will resume at 11 Eastern, which is about 13 minutes from now. 11 Eastern, listeners.
All right. Thank you, Arnold. And yes, please take us to break. All right, and welcome back from that break. We'll keep the ball rolling here, and I'm going to hand it back to our chair, Dr. Monto.
Thank you, Mike. And we are now switching to the BNT 162b2 Pfizer-BioNTech COVID-19 vaccine. And the request is for emergency use authorization for use in infants and children 6 months through 4 years of age. And we're going to hear from Dr. Gruber, Senior Vice President at Pfizer. Take it away, Bill.
Good morning. On behalf of Pfizer and BioNTech, it is my pleasure to share data supporting the BNT 162b2 request for emergency use authorization in individuals 6 months through 4 years of age. My name is Bill Gruber, and I head the Vaccine Clinical Research and Development Group at Pfizer. Today's agenda covers the topics shown here, with specific attention to coverage of the clinical safety, immunogenicity, and efficacy data, along with the assessment of benefit risk. There is a clear unmet medical need in children 6 months to less than 5 years of age for a safe and effective COVID-19 vaccine, and you've heard this discussed at length over the past 2 days. I'm going to summarize in a single slide. Less than 5-year-olds are currently the only pediatric group for whom vaccine is not available. Severe COVID-19 occurs in children less than 5 years of age, and as of May 2022, there were over 45,000 hospitalizations, with roughly half of these hospitalizations due to Omicron, with a high number of ICU admissions and deaths. The burden is comparable to influenza, as you heard from Dr. Marks, for which children are routinely immunized. Severe COVID-19 outcomes are unpredictable and can occur in healthy children. 64% of hospitalizations in children less than 5 years of age occur in those without comorbidities. COVID-19 can cause additional long-term sequelae in children. 3 to 6% of children report continued symptoms for greater than 12 weeks. And importantly, the pandemic adversely impacts development and psychosocial well-being, whether or not a child is isolated because of COVID-19 infection, because of the social distancing and other requirements that limit in-person schooling and other social interactions. The need for 3 mRNA vaccine doses to protect against Omicron-related COVID-19 is clear. Omicron is significantly more transmissible than prior variants. In adult populations, 2 doses of the current mRNA COVID-19 vaccines do not adequately neutralize Omicron. A 3rd dose increases breadth of coverage and can neutralize Omicron more effectively. Real-world data show that a 3rd dose significantly improves protection against Omicron-related symptomatic disease and severe illness. Given the high prevalence of Omicron and the emerging evidence that 3 doses of mRNA vaccine are needed against Omicron, we studied 3 doses of BNT 162b2 in children 6 months through less than 5 years of age. Pfizer-BioNTech is seeking emergency use authorization of the 3 microgram dose level of the vaccine in children 6 months through 4 years of age. The proposed indication is for active immunization to prevent COVID-19 caused by SARS coronavirus 2 in individuals 6 months through 4 years of age. The vaccine would be administered intramuscularly as 3 doses of 0.2 milliliters each. 2 doses would be administered 3 weeks apart, followed by a 3rd dose at least 8 weeks after the 2nd dose. The 3 microgram dose was chosen as it had the right balance between immune response and a satisfactory reactogenicity profile. I'm now going to share with you the clinical data that supports emergency use authorization. Here's the study overview information, which should be familiar to you, but now focuses on children 6 months to less than 5 years of age. To select the appropriate pediatric dose levels for infants, toddlers, and very young children, we carefully evaluated multiple dose levels in phase 1. We began in 5 to 11-year-olds before progressing to 64 participants in the 6-month through 4-year-old age group to achieve the right balance in safety profile and immune response. These pediatric groups represent a more vulnerable population, so it is particularly important to minimize reactions, including fever, while achieving an immune response likely to provide protection against COVID-19. We were therefore very deliberate in dose ranging. We found that nearly 19% of 2 to less than 5-year-olds who received the 10 microgram dose developed fevers after the first and second dose, and 1/3 of those fevers were severe. We were concerned that the frequency and severity of fever seen after the 10 microgram dose would likely further increase in the infant and toddler group of 6 months to less than 2 years and could be poorly accepted by parents, reducing adherence to the primary 3-dose series. In contrast, the 3 microgram dose had a much better tolerability profile combined with comparable immune responses to the SARS coronavirus 2 reference strain. Therefore, the 3 microgram dose level was advanced into phase 2/3 in the countries shown. To infer efficacy in the pediatric population in the pivotal study, immunologic non-inferiority to a 16 to 25-year-old population for whom efficacy was established was assessed in addition to safety to satisfy emergency use authorization immune response criteria. Although not required for EUA approval, COVID-19 surveillance was conducted, permitting an early evaluation of vaccine efficacy. This study schema should also be familiar to you. Children were administered 2 doses, as shown at the top, 21 days apart, and then a 3rd dose was administered at least 60 days later. Follow-up for reactions, adverse events, antibody response, and surveillance are parallel to that described for older children. Current follow-up includes 1-month post-dose 3 serology and efficacy to the data cutoff of April the 29th. Safety data as of the April 29th cutoff date follow. All safety data that I will present is in the blinded, placebo-controlled follow-up period. I will first discuss the safety data in 2 to less than 5-year-olds, and then the 6-month to less than 2-year-olds. Demographics in the 2 to less than 5-year-old age group are balanced between vaccine and placebo groups, whether gender, race, or ethnicity. Note at the bottom of the table that 12.7 to 13.7 of participants had evidence of prior or current SARS coronavirus 2 infection at the time of the first dose. Approximately 12 to 14% of participants had underlying comorbidities, including obesity. Here are the tolerability data for 2 to less than 5 years of age group. Care providers for participants with and without prior SARS coronavirus 2 infection at baseline reported local reactions by maximum severity within 7 days after each dose. These include redness, swelling, and pain at the injection site, color-coded as shown. Local reactions were mostly mild to moderate in severity, somewhat higher in the vaccine recipients compared to placebo recipients, and did not show an increase from dose 2 to dose 3. Local reactions were higher or similar in frequency and severity, and those with evidence of prior SARS coronavirus 2 infection at baseline, and all within a well-tolerated range. There were no grade 4 reactions. Care providers for participants 2 to less than 5 years of age with and without prior SARS coronavirus 2 infection at baseline reported systemic events shown in this table. Let me orient you to the slide. Dose 1, Dose 2, and Dose 3 are shown in each of the rows with the color-coded rating scales as shown. Placebo recipients are paired up with the vaccine recipients for each of the symptoms shown. Systemic symptoms solicited by electronic dialing were mostly mild to moderate. Fever, fatigue, or other symptom rates were remarkably similar to those seen in placebo recipients and much lower than those in older age groups immunized with higher dose levels. Fever rates are comparable or lower than other childhood vaccines. The low incidence of fever observed after each vaccine dose generally peaked by day 2 and declined by day 4. Only 3 or less than 0.2% of BNT162b2 participants reported fever greater than 40 degrees centigrade after dose 1 or dose 2, starting on day 4, day 2, day 4, or day 6, with all returning to normal 6 to 7 days after the dose. One of these had a presentation suggestive of a viral exanthem. None of these required hospitalization and all resolved quickly. Systemic symptoms were higher or similar in frequency and severity in those with evidence of prior SARS coronavirus 2 infection at baseline, and all within a well-tolerated range. This highly favorable tolerability profile for the Pfizer-BioNTech vaccine should be reassuring to parents and care providers. In the 2 to less than 5-year-old age group, blinded safety follow-up occurred from the time of dose 2 to dose 3, or cutoff date, for a median of 4.3 months, and for dose 3 to the cutoff date for a median of 1.4 months. As a reminder, randomization was in a 2-to-1 ratio. Unsolicited adverse events are shown here by proportion, reporting at least one adverse event with vaccine recipients in blue and placebo recipients in red. Overall, unsolicited adverse events during the blinded part of the study to data cutoff in 2 to less than 5-year-olds were comparable between vaccine and placebo recipients, and evaluation during the unblinded period did not change this assessment. Related adverse events, serious adverse events, and withdrawals were infrequent and comparable between vaccine and placebo groups. Most SAEs were gastrointestinal or respiratory infection illnesses with no imbalance between groups. 3 subjects, or 0.2%, in the BNT162b2 group withdrew from the study due to adverse events. Pyrexia considered related, status epilepticus considered unrelated, and urticaria considered unrelated. 1 participant in the placebo group withdrew to facial swelling and rash considered related. Further details are in the briefing document. There were no deaths reported in this age group. Adverse events occurring in 1% or more of participants by system organ class were comparable between vaccine and placebo recipients from dose 1 to cutoff, whether for any adverse events or the subcategories shown. Lymphadenopathy in the BNT162b2 group at the 3 microgram dose was 0.1%. This was a lower frequency of lymphadenopathy than that reported in older children and adults. I'm now going to turn attention to the safety evaluation in 6 months to less than 2-year-olds. The demographics are shown here, again with balance between the vaccine and placebo groups related to gender, race, or ethnicity. As shown at the bottom of the table, between 7.6 and 7.4% of participants had evidence of prior or current SARS coronavirus 2 infection at the time of dose 1. 4% to 6% of individuals enrolled in the trial had underlying comorbidities. Reports of local reactions by maximum severity are shown in this figure in the same way that they were shown for the older population. Local reactions in the 6-month to less than 2-year-old group with and without prior SARS coronavirus 2 infection were mild to moderate, with incidence somewhat higher in vaccine recipients. Local reactions were somewhat higher or similar in frequency and severity in those with evidence of prior SARS coronavirus 2 infection at baseline, and all within a well-tolerated range. There were no grade 4 events, and frequency remained relatively the same after each dose, again consistent with a very well-tolerated vaccine. Caregivers of participants reported E-diary systemic events by maximum severity within 7 days in this age group with or without prior SARS coronavirus 2 infection. Note that the symptoms shown in this age group differ somewhat in terms of how they are captured because of age. So shown here are fever, decreased appetite, drowsiness, and irritability, all of which were mostly mild to moderate and similar to placebo rates. Fever rates were once again comparable or lower than those observed in older children and adults. Fever rates are comparable or lower than fever rates of other childhood vaccines. Fever usually occurred by day 2 and declined by at least day 4 or sooner after each dosing with vaccine. Fever greater than 40 degrees centigrade was reported by only 3 recipients, or less than 0.1% for each dose, starting on day 1, day 2, or day 3, with all returning to normal by at least 5 to 6 days after the dose, 2 of whom had a concurrent viral infection. 1 fever greater than 40 degrees centigrade was reported by a placebo recipient after the first dose. None of these required hospitalization, and all resolved quickly. Given the similarity in reactions, much of the febrile illness in both groups may reflect viral infections common in this age group. Systemic symptoms were somewhat higher or similar in frequency and severity in those with evidence of prior SARS coronavirus 2 infection at baseline, and all within a well-tolerated range. Again, the overall incidence and low severity of systemic symptoms speaks to a favorable tolerability profile for the Pfizer-BioNTech vaccine that should be reassuring to parents and care providers. Safety follow-up, including blinded follow-up from the time of dose 2 to dose 3, or the cutoff date, was for a median of 6.3 months, and from dose 3 to the cutoff date was a median of 1.3 months. Unsolicited adverse events shown here are similar to the pattern I've described to you in the older age group. Overall, unsolicited adverse events were comparable between vaccine and placebo recipients. Related AEs, SAEs, withdrawals were infrequent and comparable between vaccine and placebo groups. Most SAEs were gastrointestinal or respiratory infection illnesses. 3 participants, or 0.3%, in the BNT162b2 group withdrew from the study due to adverse events, all related. 2 due to a fever greater than 40 degrees Celsius, one of which had a viral exam and that was considered unrelated. 1 participant withdrew due to a generalized rash on the face and trunk, and further details of these events are included in your briefing document. There were no deaths reported in this age group. Once again, adverse events rates in this group occurring in 1% or more participants by system organ class were comparable between vaccine and placebo recipients from dose 1 to cutoff. Lymphadenopathy was reported in only 2 participants, or 0.2%, in the BNT162b2 group and none in the placebo group. The frequency of reported lymphadenopathy is lower than that reported in older children and adults. Few adverse events of special interest were recorded and were of similar incidence to placebo. FDA adverse events of special interest are reported here for both age groups. Predominant categories were potential angioedema and hypersensitivity, comprising mainly urticarias and rashes. For CDC-defined adverse events of special interest, no vaccine-related anaphylaxis, no myocarditis, no pericarditis, no Bell's palsy, and no MIS-C was observed. The carefully selected dose level of 3 micrograms for the BNT162b2 vaccine was shown to have an excellent safety profile and was well tolerated in infants, toddlers, and very young children. Vaccine reactions were mostly mild to moderate and short-lived, with systemic reactions comparable to placebo. Reactions were comparable after dose 1, 2, and 3. The unsolicited adverse event profile mostly reflected reactogenicity or common childhood illnesses. The safe and well-tolerated vaccine profile of the carefully chosen 3 microgram dose should reassure parents and providers. Specifically, the vaccine provides high protection against Omicron if all 3 doses are received, and I'll share that with you shortly. The low incidence of fever and systemic reactions similar to those of placebo recipients should encourage vaccine adherence for each of the 3 doses. I will now describe immune responses in children less than 5 years of age. Demonstration of non-inferior immune response in children less than 5 years of age after 3 doses compared to immune responses in 16 to 25-year-olds after 2 doses was judged by the FDA as sufficient to meet immunologic success criteria for emergency use authorization. Immunobridging criteria in the 2 to less than 5-year-olds were met for both GMR and seroresponse, which infers vaccine effectiveness in this age group. Shown here are the SARS coronavirus 2 neutralization assay titers to the reference strain post-dose 3 in children 2 to less than 5 years of age in the light blue after 3 doses, compared to those 16 to 25 years of age in the darker blue after 2 doses, with the geometric mean ratio shown on the right-hand side. The median dosing time between dose 2 and dose 3 was 10.7 weeks. The geometric mean ratio observed was 1.30 and importantly had a lower bound of 1.13, thus well above the 0.67 success criteria required by the FDA. This was also true for the immunobridging criterion relying on seroresponse. I draw your attention to the difference in percent responders on the right-hand side, which is 1.2% between the 2 to less than 5-year-olds and the 16 to 25-year-olds, with a lower bound of minus 1.5%, which is well above the minus 10% non-inferiority success criterion. I now turn to the 6-month to less than 2-year-olds. Immunobridging criteria were met for both GMR and seroresponse, which infers vaccine effectiveness in this age group as well. The median dosing time between dose 2 and dose 3 was 12.9 weeks. The scheme is the same, and I draw your attention to the right-hand side of the slide where the geometric mean response was 1.19 with a lower bound of 1, well above the 0.67 required success criteria. Immunobridging criteria were met for the seroresponse with 100% of children less than 2 years of age responding, and a difference in percent of responders of 1.2% with a lower bound of minus 3.4, again well above the minus 10% required success criterion. So for both younger children as well as the older children, immunobridging criteria were met. Given that most current COVID-19 cases are caused by Omicron, we also evaluated the ability of 2- and 3-dose immune sera from 6 months to less than 5-year-olds to neutralize Omicron compared to sera from adults. In this graph, the older comparator group is a 24 to 74-year-old adult sentinel cohort from the licensure trial that has been used throughout development to evaluate antibody response to emerging variants. Immune responses to Omicron, shown in pink, were compared to those of the reference vaccine strain, shown in blue, using a plaque reduction neutralization assay. GMTs are shown on the y-axis and age groups are shown left to right. As you can see, we see comparable immune responses across all 3 age groups to the reference strain and Omicron. However, as reported by several groups, Omicron responses after only 2 doses are low and uniformly so across the pediatric and adult groups. Low Omicron neutralization titers after 2 doses seem to correlate with lower efficacy after 2 vaccine doses for Omicron. However, this picture changes when serum samples after 3 doses are being evaluated. In this data set, an adult comparator group was used that received the 3rd dose at a similar time interval after the 2nd dose as the pediatric group at about 11 to 13 weeks. Neutralizing antibody responses were measured using a fluorescent focus neutralization assay. This data has been submitted to the FDA and is a supplement to the briefing document. Left to right, for 6 months to less than 2-year-olds and 2 to less than 5-year-olds, the Omicron-specific neutralization titers after 3 doses are far higher than those on the prior slide after 2 doses. And the Omicron-specific titers are very similar across age groups. Most importantly, the pediatric group titers are essentially the same as in the adult group, predicting that similar efficacy as shown in adults could likely be observed for the 6-month to less than 5-year-old age group. I will shortly share with you descriptive data showing observed post-dose 3 efficacy matching this prediction. So here are the immunogenicity conclusions in 6 months to less than 5-year-olds. All immunobridging criteria post-dose 3 in young children required for an emergency use authorization were met for both age groups, inferring effectiveness. Omicron neutralization titers were low after 2 doses for pediatric and adult cohorts, but increased substantially after a 3rd dose in the 6-month to less than 5-year-olds to similar levels observed in adults. Thus, as has been observed in other populations, a 3rd dose is required also for the 6-month to less than 5 years of age group to ensure a more robust protection against COVID-19 due to Omicron. So what have we learned from the adult and pediatric experience about the potential for BNT162b2 efficacy against COVID-19? Here is a summary of observed efficacy data in the blinded follow-up period that, while not required for an EUA, supports the importance of a 3rd vaccine dose to protect children less than 5 years of age against Omicron, just as a 3rd dose is important to protect older children and adults against Omicron. It's important to note that nucleic acid amplification testing was defined either based on a central laboratory determination or an acceptable test in a local laboratory for all cases. In addition, it's important to remember that the follow-up for this population after the second dose was greater than 4 months for 2 to less than 5-year-olds and greater than 6 months for 6 months to less than 2-year-olds. Details, including 95% confidence intervals, are in your briefing document, and the 95% confidence intervals for Omicron after the third dose are shown on the next slide. Efficacy is shown on the y-axis and by age, 6 months to less than 5 years, on the left, with the age groups displayed progressively to the right. Efficacy against the Delta variant is shown in blue, and efficacy against the Omicron variant is shown in pink. Please note that the Delta surge ended prior to the third dose, so no children were exposed to Delta after the third dose, marked as NA. You can see efficacy against Delta post-dose 2 in the evaluable population without evidence of infection prior to 7 days post-dose 2 of 70.2% was observed in 6 months to less than 5-year-olds, shown on the left, and moving left to right, 56% in 2 to less than 5-year-olds and 91.6% in 6 months to less than 2-year-olds. This is consistent with a high level of efficacy against Delta after 2 doses of vaccine and comparable to that observed in older children and adults. The emergence of Omicron presents a new challenge. Shown in pink on the left, you can see that for children 6 months to less than 5 years of age post-dose 2, efficacy was 21.8% in the 6 month to less than 5-year-old age group, and correspondingly as shown for the other subgroups. This is consistent with the poor antibody response after the second dose that I shared with you, and is consistent with lower efficacy against Omicron in older children and adults after 2 doses compared to other variants like Delta. Now look what happens after the third dose. Cases post-dose 3 in this clinical study occurred after February 7, 2022, and were confirmed to be Omicron. This confirmatory data is being submitted to the FDA. Efficacy in the all-available population against Omicron rises to 80.3% overall, and correspondingly so for the respective age subgroups. This descriptive observed efficacy well exceeds the original FDA guidance for an efficacy point estimate of at least 50%. This is consistent with the higher antibody response seen after the third dose against Omicron, like that of older children and adults, and consistent with corresponding higher efficacy in older children and adults after a third dose against Omicron. This table displays details of the high descriptive 80% efficacy observed after the third dose during a period when Omicron was predominant. Note that the large N in this table represents the population followed during the placebo-controlled blinded period of the trial up to the data cutoff of April the 29th. And note that the trial was randomized 2-to-1 vaccine to placebo. Whether we're talking about the 6 month to less than 5-year-olds or for the subgroups, high efficacy was observed in the course of this trial with 80.3% efficacy shown in the overall age group with a 95% confidence interval lower bound of 13.9%. 82.3% and 75.5% efficacy were observed in the respective age subgroups with larger confidence intervals, of course, because these subgroups are smaller and individually have a smaller number of cases. So what can we conclude about efficacy? As demonstrated in other pediatric and adult age groups, 2 doses of BNT162b2 are protective against variants of concern such as Delta, but do not provide adequate protection against Omicron. As demonstrated in other pediatric and adult age groups, a third dose is necessary to provide high protection against Omicron. In addition to 6 months of follow-up as part of the current clinical trial, ongoing and active pharmacovigilance and pharmacoepidemiology will continue with a focus on any expanded pediatric populations receiving vaccine. These include the pharmacoepidemiology studies for ages 6 months and up, and you can see that 5 studies are noted here. No myocarditis was noted in the clinical trial of children less than 5 years of age, and this rare event does appear to be less of a risk for 5 to 11-year-olds. However, evaluation for such a rare event will be expanded into this age group as part of routine pharmacovigilance. Proactive risk mitigation will continue, including labeling educational material and vial differentiation with a maroon top, and pharmacovigilance will continue as shown and be consistent with pharmacovigilance in older children and adults already underway. The potential benefits of vaccinating children 6 months to less than 5 years of age outweigh the known potential risk. This age group of 6 months to less than 5 years of age is currently unprotected. Protection against COVID-19 is critical, particularly in light of the unpredictability of potential new waves and emergence of new variants of concern. Available safety, immunogenicity, and efficacy information support a highly favorable benefit-risk profile for administration of 3 doses of BNT162b2 at 3 micrograms to children less than 5 years of age. Overall, given the favorable benefit-risk profile, Pfizer-BioNTech requests emergency use authorization of BNT162b2 for active immunization of individuals 6 months through 4 years of age, administered intramuscularly as a 3-dose series. Pfizer and BioNTech wish to thank the clinical trial participants, sites, investigators, CRO, our partners and their staff, and the FDA guidance to assess this urgent medical need. I and my Pfizer colleagues will now be happy to take questions.
Thank you, Dr. Gruber. Dr. Pergam is next, followed by Dr. Gans. Dr. Pergam.
Thanks, Dr. Gruber, for that presentation. I was curious, we haven't seen much of the data related to the dose finding in the initial phase 1 study. I'm curious if you can describe a little bit about whether there was a difference in immunogenicity between the 3 microgram versus 10 microgram. Is there a dose dependency in that particular comparison? I'm just curious based on differences in dosing of the vaccine.
Thanks for that question. Obviously, as I said, we paid strict attention to finding just the right dose that gave us the appropriate immune response after 3 doses. There are really 2 lines of evidence that support the basis for the dosing decision. One is the reactogenicity that I already shared with you. But the second one is really, if we can bring up the slide with the pink bars that speaks to the Omicron response. You may recall that while that's coming up, that we made decisions at the time of phase 1 based on comparisons to an adult population that gave us reassurance that for the reference strain we were likely to meet non-inferiority, right? Because that's the criteria for licensure. But in addition to that, so if we can put slide 3 up, this is really the most compelling information, that having chose the 3 microgram dose and giving it as 3 successive doses, we now essentially nearly equal the sort of response that one sees in adults for which we know we have good protection against Omicron. So I think that confirms for us with the reactogenicity profile that I shared you and this finding that this is the right dose to provide protection. And again, although it's it's early, the 80% efficacy that we're seeing essentially matches what you would expect based on this type of response.
Okay. Again, Mike, just as a piece of clarification, did you see a difference in the 3 microgram versus 10 microgram in terms of antibody responses? It's just a clarification question.
The nature of the antibody responses were both above the responses that we saw in adults, but combined with the reactogenicity profile and knowing that for the reference strain we would be in a position to potentially meet non-inferiority, we chose that strain because we chose that dose because we again wanted to be confident that the vaccine would be accepted. Obviously, we already know that in older individuals, 30% of children are not getting the vaccine. There could be a lot of reasons for that, but one of them is the reactogenicity. So combined with that, and again, the data that I'm showing you here, it seems pretty clear that the 3 microgram dose is the right dose. And followed by obviously the phase 3 data that we have on reactions, which are very comparable to what we see in placebo recipients.
Okay, thanks.
Thank you. Moving on, Dr. Gans, followed by Dr. Portnoy.
Thank you for that presentation. I had a follow-up question maybe to Steve's question regarding the dosing, and I was looking at the slightly lower immune responses in the 2 to 5-year-old to the current dosing, and I'm just wondering, are there additional studies that actually may be looking at differing dosing and splitting these groups? As we know, it's a very large immunologic group, and there may be some nuances to how the toddlers or the preschool group is different than the infant group. So that's one question. I would also like you to talk a little bit about your breakthrough disease and see if there was any differences in the severity of disease in those who had received vaccine versus those who were in the placebo group. I realize hospitalization wasn't something, so that's not really what I'm asking. I'm asking about the disease profiles in those two groups.
Let me take your first question first about the nature of splitting the groups. I think the approach that we've taken is very consistent with every other age group in terms of dealing with the pediatric population. So for 12 to 17-year-olds, they get a 30 microgram dose. For the 5 to 11-year-olds, they get 10. And for now the 6-month to 5-year-olds, they receive 3 micrograms. And again, based on what we saw in the older children, where we had 19% severe fevers in the phase 1 group, we really don't feel comfortable expanding on that dose, and we don't need to, because we now see with the 3-dose series, which I think most people now agree, and we heard from Paul Offit yesterday, the importance of a 3rd dose in an Omicron era, that this is the right dose to immunize children to protect against Omicron. For the second piece, the nature of severity of the illness, we've actually included in your briefing document, and there was really no difference if we looked at all the cases. So I'm talking about based on the number of symptoms that they had in terms of breakthrough compared to placebo, whether it was 1, 2, 3 or more symptoms. There really was not a dime's worth of difference between the respective groups, between those that break through that receive vaccine versus placebo. So that gave us confidence in that larger population of cases that we have that does not appear to be an issue.
It also doesn't seem to, if the disease is the same in the placebo group, it didn't show a necessary advantage of the vaccine, I guess was what I was
guess the advantage of the vaccine is you're preventing the infection in the first place, right?
I get that.
That's the big advantage, right? But the good news is it doesn't seem to make the disease worse if, in fact, you've received the vaccine. And I think that's an important thing and why we looked at it.
Thank you. Dr. Portnoy, followed by Dr. Cohen.
Thank you. I guess I'm a little bit confused about this dosing in terms of micrograms, because your dosing is 3 micrograms, the Moderna dosing is 25 micrograms. Clearly, we're thinking in terms of micrograms the way we would think of proteins as a way of inducing an immune response, and yet the purpose of mRNA is to induce protein production. So is your mRNA just more efficient at making cells produce protein, or how should we think of micrograms in terms of the amount of spike protein that's produced by the cells? Can you kind of clarify that?
Yeah, I'll leave to Moderna to describe the nature of how they address their vaccine dosage. But I think, obviously, we don't have a complete understanding of the nature of the way that the vaccine works in terms of producing an immune response. So you have to go by the results. And the results are that in the setting of giving a 3 microgram dose, we had low reactogenicity compared to placebo. And after a 3rd dose, just as in adults at higher doses, we're getting an immune response that's comparable. It may well be that children, we've seen certainly that we're able to go down to a lower dose in children, and the expectation is perhaps they have a more robust response. That seems to be the case based on giving a 10 microgram dose to 5 to 11s and 3 micrograms to younger.
But have you ever measured the amount of protein that's produced as a result of the mRNA and how many cells are producing it and how persistent that production is for a given microgram of mRNA?
That's a pretty broad question.
Yeah.
I think that's obviously, an interesting question to better understand the mechanism, and I would say it's somewhat academic in the setting of what we're trying to achieve here in terms of getting an immune response and a safety profile that's satisfactory, but worthwhile for people to pursue.
Okay, thank you.
Let me just, Dr. Jansen, the head of Vaccine Research and Development, would like to make a comment about that last question.
Very brief.
Thank you, Bill. I think one important consideration for the answer to the question that was just posed is that the two mRNA vaccines are not created equal. They're actually very different vaccines. They use the same platform. They have different formulations. And so I think that's important to recognize. And the second piece is that we, of course, have optimized the vaccine for optimal expression of the antigens in cells. And if you ask the question, is there a large number of protein molecules expressed in the cells, the answer is yes. Thank you.
Thank you both. Dr. Cohen, final question.
Thank you. So I have this is maybe a multi-part question, but I'm wondering how you can be so sure of both your immunogenicity and VE estimates in the setting of the requested interval that you've requested between 2 and 3 doses is actually different than any of the is actually very different than the median in which your recipients received that third dose. So how can you be sure that they would have a similar response 8 weeks after the first dose? And that sort of goes back to, this time period between the second dose where there appears to be very little to no effectiveness and the third dose would essentially mean that these kids would not be protected at all for an additional 8 weeks. So I'm trying to sort through both, you looked at effectiveness in kids who were vaccinated at a longer period after that second dose. And if you have any kids or any data on immunogenicity after 8 weeks.
So thanks for the question. I think where we take a great deal of comfort is sort of comparing like to like. So the nature of comparing this 11 to 13 week comparison in the older adult population to the immune response we're seeing in the younger sort of translates to the likelihood that you're going to get similar efficacy with that interval. Likewise, I think it's reasonable to assume that if the interval were lower, you would compare to what we saw in adults as well. I think there were a fair number of individuals that did actually get the vaccine in the 8 to 13 week period that are obviously being monitored as part of our ongoing assessment of efficacy, and perhaps we'll learn something from that, particularly as time goes on and we have longer follow-up.
So, but to clarify, we don't actually do we have data on adults that are immunocompetent that were vaccinated 8 weeks after their second dose?
We, in terms of data, in terms of 8 weeks, I'm not sure that we actually have data that we've analyzed at 8 weeks.
Right. So essentially what you're saying, and I hear your confidence, which is great, but what you're asking for, this 8 week interval, is not the same and can't be compared to adults or to the kids, to many of the kids who are in your study.
I guess, what has to, and Dr. Cohen, I appreciate the question. I think what has to be balanced here is a reasonable expectation that we'll have some level of efficacy, certainly seeing that we have 80% at 11 to 13 weeks, and the need to get children vaccinated as quickly as possible to essentially achieve that efficacy. That was part of the reason, as you know, that for our primary series, we looked at 21 days. We wanted to narrow that interval what was reasonably possible to have enough maturation of immune response that you get a good response after the second dose. If we lengthen out now the period of time after the third dose, and we're all hearing, and the data supports that you need a third dose for Omicron, you're essentially trading off a theoretical issue that maybe I'm not going to get quite as good efficacy for the notion of a child, having that period of exposure before they can get that third dose. So I think that is the sort of thing that has to be weighed.
Thanks.
Okay, thank you, Dr. Gruber. We're moving on now to the FDA presentation on the efficacy and safety of the Pfizer-BioNTech vaccine. Dr. Susan Wollersheim in the Clinical Review Branch, FDA, will be giving us the presentation. Dr. Wollersheim.
So much, Dr. Monto. Can you hear me okay?
We can.
Thank you. And Mike, if you could add my notes, that would be great. Fantastic. I see them now. Thank you so much. I'm Susan Wollersheim, a medical officer in the Division of Vaccines and Related Products Applications at the FDA. I will be presenting the FDA's review of the effectiveness and safety of the Pfizer-BioNTech COVID-19 vaccine in children 6 months to 4 years of age, submitted under an Emergency Use Authorization Amendment, or EUA. And I'd like to start by acknowledging so many contributions from so many colleagues across the FDA. Thanks so much. To outline my presentation for you, I will start by providing the regulatory background of the product and the study design, followed by a brief review of the Phase 1 dose selection, then the Phase 2-3 immunogenicity, descriptive efficacy, and safety results, followed by the Pharmacovigilance Plan and a summary of the benefits and risks for this product. To begin, the Pfizer-BioNTech COVID-19 vaccine is based on the SARS-CoV-2 spike glycoprotein S antigen derived from the Wuhan strain encoded by RNA and formulated in lipid particles. This slide shows a summary of the various age groups, dose levels, regimens, and current authorizations and approvals for the primary series of this product. The EUA under discussion today is listed at the bottom of the slide and is intended to support use of a 3-dose primary series of the Pfizer-BioNTech COVID-19 vaccine at the 3 microgram mRNA dose level. The FDA has issued related EUAs previously as listed at age-appropriate dose levels as a 2-dose primary series and a 3rd primary series dose for certain populations. In August of 2021, the Pfizer-BioNTech COVID-19 vaccine was approved under Biologics License Application under the proprietary name Comirnaty for use in individuals 16 years of age and older. And this slide presents some key features of the 2 prior pediatric EUAs issued for the Pfizer-BioNTech COVID-19 vaccine in May of 2021 for adolescents 12 through 15 years of age and October 2021 for children 5 through 11 years of age. The EUA issued for children 5 through 11 years of age is for the 10 microgram dose level, while the 30 microgram dose level is authorized for use in adolescents 12 through 15 years of age. Data from clinical studies submitted to support the EUAs for both age groups included similar safety endpoints, immunobridging approaches, and descriptive efficacy analyses. The safety database for vaccine recipients and percentage of participants with 2 months or more of follow-up at the time of each EUA data cutoff are shown. And as you will see from the rest of the presentation, this EUA request for children 6 months through 4 years of age has similar features. Now we'll move on to the study design. Study C4591007 is an ongoing Phase 1-2-3 randomized, blinded, placebo-controlled study to evaluate the safety and effectiveness of BNT162b2 in children 6 months through 11 years of age and was the basis for the EUA issued for children 5 through 11 years of age. The focus of this EUA request is the remaining participants 6 months through 4 years of age, also enrolled in the study. My slides are moving, sorry. Okay, so for Phase 1, a 2-dose primary series of the vaccine was evaluated in U.S. children who were not at high risk for SARS-CoV-2 exposure, did not have medical conditions that represented risk factors for severe COVID-19, and did not have evidence of prior SARS-CoV-2 infection. Dose levels of 3 and 10 micrograms were evaluated in an open-label manner for each age group, starting with the older age group and based upon safety evaluation and recommendation by the Internal Review Committee. Selection of the 3 microgram dose level for both age groups was driven by reactogenicity and supported by immunogenicity obtained at 7 days post-dose 2. Phase 2/3 of study C4591007 is being conducted in the United States, Finland, Poland, and Spain. This portion of the study did not exclude children with a history of SARS-CoV-2 infection, children with known HIV, hepatitis B, hepatitis C, or stable pre-existing chronic disease. Participants were randomized 2-to-1 to receive 2 doses of vaccine or saline placebo 3 weeks apart. And immunogenicity was assessed in a subset of participants at 1 month post-dose 2 to infer effectiveness as the primary endpoint. Following analysis of the post-dose 2 safety and effectiveness data, a third primary series dose was added for participants 6 months through 4 years of age, at least 8 weeks after dose 2, in Protocol Amendment 6. Approximately 4,500 total participants have been enrolled at the time of this data cutoff, and enrollment is ongoing to expand the safety database. Immunogenicity at 1 month post-dose 3 was assessed in a subset of participants, and efficacy data was obtained through continuous surveillance for potential cases of COVID-19. There were 2 circumstances for participants to be unblinded during the study, and the first was if they turned 5 years of age and became eligible to receive vaccine as available under the EUA from October of 2021, and the second was the planned unblinding at the 6-month post-dose 2 visit in the original protocol, prior to the addition of the third dose to the primary series. So subjects randomized prior to Protocol Amendment 6 were unblinded 6 months post-dose 2 and offered vaccine if they originally received placebo. The first placebo crossover occurred in November of 2021. Subjects randomized after implementation of the Protocol Amendment 6 will be unblinded at their 6-month post-dose 3 visit and offered vaccine if they originally received placebo. The notable implications of unblinding during this study are that the descriptive efficacy analyses include only blinded participants. However, the safety analyses do include all participants who received any study intervention, regardless of those who are blinded or unblinded. This slide outlines the study objectives and endpoints. Immunogenicity was evaluated 1 month post-dose 3, and analyses were conducted by age group, with 2 primary endpoints of geometric mean titers and seroresponse rates tested sequentially. Efficacy was evaluated with continuous surveillance for potential cases of COVID-19, and safety data was collected from all participants who received study intervention as follows. Safety analyses included solicited local and systemic reactions or reactogenicity for 7 days after each vaccination via e-diary. Unsolicited adverse events were collected within 30 minutes after each dose, which were considered immediate adverse events, and from dose 1 through 1 month after each dose. Serious adverse events will be collected from dose 1 through 6 months after dose 3 or the data cutoff. The effectiveness of the Pfizer-BioNTech COVID-19 vaccine is being inferred by comparing neutralizing antibody responses against the Wuhan-like strain obtained 1 month post-dose 3 in the pediatric age group separately, compared to a subset of participants 16 through 25 years of age, enrolled in the vaccine efficacy study C4591001, in which the overall vaccine efficacy was 91.2% in participants 16 through 55 years of age. Participants in each immunogenicity analysis population did not have evidence of prior SARS-CoV-2 infection. Immunobridging endpoints and statistical success criteria will be discussed in the next 2 slides. This slide shows the first immunobridging analysis based on geometric mean titer, or GMT. The SARS-CoV-2 neutralizing antibody titers obtained 1 month post-primary series are being compared in participants without evidence of prior SARS-CoV-2 infection. The GMT ratio compares each pediatric age group to the young adult age group from study C4591001 with success criteria that the lower limit of the two-sided 95% confidence interval is greater than 0.67 and the point estimate of the GMT ratio is greater than or equal to 1. The second immunobridging analysis is based on seroresponse rate, which is defined as the percentage of participants with a greater than or equal to 4-fold rise from baseline. The success criteria is a lower limit of the 95% confidence interval for the difference in seroresponse rates being greater or equal to -10%. Both immunobridging analyses had to meet success criteria for overall success of the primary endpoint. And this slide provides the case definitions for protocol-defined symptomatic COVID-19 and severe COVID-19 used for the vaccine efficacy analyses. Symptomatic COVID-19 was defined as the presence of at least one of the listed symptoms on the left side of the slide, as well as a confirmed SARS-CoV-2 PCR positive test during or within 4 days of the symptomatic period. Severe COVID-19 includes the symptomatic COVID-19 case definition with at least one of the listed criteria on the right side of the slide, such as abnormal vital signs, various levels of respiratory and systemic illness, ICU admission, or death. Vaccine efficacy was a secondary objective planned after 21 confirmed cases had been accrued across both age groups and conditional on successful immunobridging. This slide shows the various analysis populations for each age group for which you will see results today. The numbers of vaccine and placebo recipients listed reflects the 2-to-1 randomization, and the first row shows the total numbers of participants who received any dose of study intervention. There are notably smaller numbers of participants in the dose 3 efficacy analysis population shown in the bottom row of the slide due to unblinding and attrition. For the 6 through 23-month age group, a total of 715 original vaccine recipients and 377 original placebo recipients were unblinded during the study conduct. And for the 2 to 4 year age group, a total of 842 original vaccine recipients and 424 original placebo recipients were unblinded. This slide presents the demographics and baseline characteristics of subjects 6 through 23 months of age. These demographics are also comparable to the immunogenicity and efficacy populations, which are subsets of this overall safety population. The treatment groups were balanced in terms of demographics and baseline characteristics. The median age was 16 months. 7 to 8% were positive at baseline for SARS-CoV-2 status. The majority of subjects were white, and 14% identified as Hispanic. Participants were enrolled in 4 countries, the U.S., Spain, Finland, and Poland, with the U.S. contributing most participants. Comorbidities were reported in 4 to 6% of participants, including asthma, cardiovascular disease, and congenital heart disease. And this slide shows the demographics of the safety population for the older age group, which was generally comparable to the younger age group. And as a reminder, the immunogenicity and efficacy populations are also subsets of this overall safety population. The treatment groups were balanced in terms of demographics and baseline characteristics. The median age was 3 years. Comorbidities were reported in 12 to 14% of participants, including asthma, neurologic disorders, and congenital heart disease. Obesity was present in approximately 7%. And of note, there were no HIV-positive subjects enrolled into the study for these age groups. We will now move on to the immunogenicity data. Here you see the results for the GMT primary endpoint in children 6 through 23 months of age. Among participants in the evaluable immunogenicity population without prior evidence of SARS-CoV-2 infection, the ratio of GMTs was 1.19, which met the success criteria for immunobridging, as the lower bound of the two-sided 95% confidence interval for the ratio was greater than 0.67, and the point estimate was greater than or equal to 1. Results for subgroup analyses of the GMTs in children 6 through 23 months of age by baseline SARS-CoV-2 status are displayed here. The definition of baseline SARS-CoV-2 status is based on results of N-binding antibody and SARS-CoV-2 PCR tests obtained prior to dose 1. Participants with positive baseline SARS-CoV-2 status had numerically higher GMTs compared to those negative at baseline, which is consistent with immunogenicity results observed in the older age group. The numbers of baseline positive participants, though, was limited to 6 in the vaccine group and 8 in the placebo group. The baseline negative group may also include participants that became infected after dose 1 and before 1 month post-dose 3 of note. Results for the seroresponse rate immunobridging endpoint in children 6 through 23 months of age are displayed here. Among participants in the evaluable immunogenicity population without evidence of SARS-CoV-2 infection, the percent difference in seroresponse rates was 1.2, which met the success criteria as the lower bound of the two-sided 95% confidence interval was greater than negative 10%. And here we'll move on to the older age group. The results for the GMT immunogenicity endpoint for children 2 through 4 years of age are displayed here. Among participants in the evaluable immunogenicity population without evidence of SARS-CoV-2 infection, the ratio of GMTs was 1.3, which met the success criteria for immunobridging as the lower bound of the two-sided 95% confidence interval was greater than 0.67 and the point estimate was greater than or equal to 1. Results for subgroup analyses of the GMTs in children 2 through 4 years of age by baseline SARS-CoV-2 status are displayed here. Participants with positive baseline SARS-CoV-2 status had numerically higher GMTs compared to those negative at baseline, which is consistent with immunogenicity results observed in older age groups. There were small numbers of baseline positive participants, as there were only 13 in the vaccine group and 8 in the placebo group. And again, this does not account for participants that became infected after dose 1, but before 1 month post-dose 3. Results for the seroresponse rate immunogenicity endpoint for children 2 through 4 years of age are displayed here. Among participants in the evaluable immunogenicity population without prior evidence of SARS-CoV-2 infection, the percent difference in seroresponse rates was 1.2, which met the success criteria as the lower bound of the two-sided 95% confidence interval was greater than negative 10%. An additional exploratory immunogenicity analysis was performed and randomly selected subsets of participants from each age group without evidence of prior SARS-CoV-2 infection. Neutralization of the reference strain, Delta, and Omicron variants were evaluated using a non-validated assay measured before dose 3 and 1 month after dose 3. The neutralizing titers at 1 month post-dose 3 are displayed here with the geometric fold rise from the baseline titer also shown. These results indicate that the third vaccine dose elicits neutralizing titers against all 3 SARS-CoV-2 viruses. And notable is that the Omicron neutralizing titers are approximately 6-fold lower than neutralizing titers against the Delta variant and reference strain. We'll now move on to the descriptive vaccine efficacy data. This slide provides the blinded follow-up time after dose 3 for each age group in the dose 3 all-available efficacy population. Just over 30% of participants had follow-up duration of 2 months or longer. The median efficacy follow-up time after dose 3 for the younger age group was 1.3 months or 5 weeks, and the older age group, the median follow-up time after dose 3 was 1.4 months or 6 weeks. For participants 6 through 23 months of age, the median timing of dose 3 administration after dose 2 of vaccine was 16 weeks, with a range of 8 to 31.9 weeks. For placebo, the median was 15.9 weeks with a range of 8 to 35 weeks. For participants 2 through 4 years of age, the median timing of dose 3 administration after dose 2 of vaccine was 11 weeks with a range of 8 to 34.1 weeks, and if placebo was 11 weeks with a range of 8 to 31.1 weeks. Of note, the following vaccine efficacy estimates are preliminary as a total of 21 cases were not accrued, and the analyses are not presented in the protocol-specified efficacy analysis population. In participants 6 through 23 months of age, with and without evidence of SARS-CoV-2 infection prior to 7 days after dose 3, the observed vaccine efficacy against confirmed COVID-19 occurring at least 7 days after dose 3 was 75.6%. with a lower limit of the 95% confidence interval of negative 369.1, based on 1 case in the vaccine group compared to 2 in the placebo group. Again, vaccine efficacy post-dose 3 cannot be precisely estimated due to the limited number of cases accrued during blinded follow-up, as reflected by the very wide confidence interval seen here. Vaccine efficacy in the dose 1 all-available efficacy population for any confirmed COVID-19 case that occurred after dose 1 is displayed here. To show the progression of vaccine efficacy following dose 1 in the top row, which was approximately 14%, the vaccine efficacy estimates varied between dose 2 and 3. And following dose 3, the preliminary vaccine estimate appears improved, but again has a very wide confidence interval and a lower limit of negative 370.1. Here is a cumulative incidence curve for participants 6 through 23 months of age for confirmed COVID-19 cases occurring at any time after dose 1. COVID-19 disease onset appears to occur similarly for both vaccine and placebo recipients until closer to the data cutoff, at which point the curves begin to diverge. Dose 2 was administered 3 weeks after dose 1, so that's easy to track on this curve near the 21-day mark on the bottom axis. But there's no clear, and so you don't see a clear effect of dose 2 on the incidence of cases between the treatment groups. What's a little more difficult to identify is the time point for dose 3 because there are highly variable dosing intervals between doses 2 and 3 with a median interval of 112 days and a range of 56 to 245 days. We'll move on to the older age group here. Participants 2 through 4 years of age with and without evidence of SARS-CoV-2 infection prior to dose 3. The observed vaccine efficacy against confirmed COVID-19 occurring at least 7 days after dose 3 was 82.4%, with a lower bound of the 95% confidence interval of negative 7.6. This is based on 2 COVID cases in the vaccine group compared to 5 in the placebo group. The vaccine efficacy post-dose 3 cannot be precisely estimated due to the limited number of cases accrued during blinded follow-up, as reflected by the wide confidence intervals associated with these estimates. Here you see the vaccine efficacy in the dose 1 all available efficacy population for any confirmed COVID-19 case that occurred after dose 1. This shows the progression of vaccine efficacy following dose 1 in the top row, which was 32.6%, and then the vaccine efficacy estimates between dose 2 and 3 varied. And following dose 3, the preliminary vaccine efficacy estimate appears approved, and again with a very wide confidence intervals and a lower limit of negative 8. And here is the corresponding cumulative incidence curve for participants 2 through 4 years of age for confirmed COVID-19 cases occurring any time after dose 1. COVID-19 disease onset appears to occur similarly for both vaccine and placebo groups until around the midway point of the curve, where they begin to diverge slowly. Dose 2 is administered about 3 weeks after dose 1, so you can track that on the lower axis near the 21-day mark. And you don't see a clear benefit of dose 2 on the incidence of cases. What's more difficult, again, is the time point for dose 3, because there are highly variable dosing intervals between doses 2 and 3, with a median of 77 days and a range of 42 to 239 days. Additional considerations about the descriptive efficacy analyses are provided here. All cases occurred during a time period when Omicron was the predominant circulating variant. There is 1 hospitalization for severe COVID-19 disease in a 2-year-old vaccine recipient, which occurred 99 days after dose 2. The other 7 severe cases that occurred any time after dose 1 met severe criteria because of 1 vital sign measurement, which was not considered clinically significant, and they were not hospitalized for COVID-19. Interpretation of these vaccine efficacy estimates is limited by the small number of confirmed cases and the short duration of follow-up after dose 3, which was only 35 days for the participants in the younger age group and 40 days in the participants in the older age group. We will now move on to the phase 2-3 safety data. This slide provides the total follow-up time, combining blinded and open label follow-up after dose 3 in the safety population. The blinded follow-up time durations are the same as the dose 3 efficacy populations described earlier. The median total follow-up time after dose 3 for both age groups was 2.1 months. Approximately 60% of participants 6 through 23 months of age and 57% of participants 2 through 4 years of age had more than 2 months of total follow-up time. Here we see the analyses for immediate adverse events, and there are very few immediate adverse events defined as an adverse event reported within 30 minutes of any vaccine dose. The events reported were consistent with local solicited adverse events, and there were no anaphylaxis events reported within 30 minutes of vaccine. Here you can see the frequency of local reactions in children 6 through 23 months of age, including injection site tenderness, redness, and swelling. Pain at the injection site was reported most commonly, followed by redness and then swelling. Local reactions generally occurred at similar frequencies after any dose, with slightly less frequency with subsequent doses. Median day of onset and duration was 1 to 2 days for all doses and treatment arms. There are very few severe reactions, with 0.1% reporting tenderness at the injection site post-dose 2 and 0.3% reporting redness post-dose 3. Here are the frequencies of local reactions in children 2 through 4 years of age. My slides are jumping. Let me go back to that slide. Go. Local reactogenicity for the older age group included injection site pain, redness, and swelling. Pain at the injection site was reported most commonly, followed by redness and swelling. And local reactions generally occurred at similar frequencies after each dose. Local reactions graded as severe were very uncommon, seen in only 0.1% of participants for redness at injection sites, followed by dose 1 and 2. The median day of onset and duration was 1 to 2 days for all doses and treatment arms. Solicited systemic reactions in vaccine recipients occurred at similar frequencies after any dose, with decreasing frequency with subsequent doses. The median day of onset and duration was 2 days for all doses and treatment arms. Severe systemic reactions were reported by 0.6% or less of participants following any dose. The percentage of participants 6 through 23 months of age who reported e-diary data and who were also baseline SARS-CoV-2 positive was 7.5%. Subgroup analyses of solicited adverse reactions by baseline SARS-CoV-2 status showed similar reactogenicity profiles. Of note, 3 vaccine recipients reported a fever greater than 40 degrees Celsius, as noted in the briefing document. Here is the first part of the systemic solicited reactions for children 2 through 4 years of age. Of note, these are different than for the children the younger age group, based on appropriate age-appropriate reporting. Solicited systemic adverse reactions in the vaccine recipients generally occurred at similar frequencies after any dose, or with decreasing frequency with subsequent doses. The median day of onset was 1 to 2 days, and the median duration was also 1 to 2 days for all doses and treatment arms. Of note, vaccine recipients 2 through 4 years of age reported a fever of greater than 40 degrees Celsius, as noted in the briefing document as well. Here's a continuation of the solicited systemic reactions for participants 2 through 4 years of age. Solicited systemic adverse reactions in the vaccine recipients generally occurred at similar frequencies after any dose, or with decreasing frequency with subsequent doses. The percentages of participants who use antipyretic or pain medication within 7 days of each study intervention are shown as well. The percentage of participants 2 through 4 years of age who reported e-diary data who were also baseline SARS-CoV-2 positive was 12.6%. Subgroup analyses of solicited adverse reactions in each age group by baseline SARS-CoV-2 status showed similar reactogenicity profiles. The frequencies of unsolicited adverse events by age group were shown there. Let me see if I can go back. Okay, the most commonly reported adverse events were consistent with solicited adverse reactions or events which commonly occur in this age group, such as infections and injuries considered not related to the study intervention. The events that were considered related to vaccine included lymphadenopathy and hypersensitivity, as has been previously described. Analyses for the adverse events of clinical interest are displayed for both age groups. Here, the FDA conducted standardized Madeira queries or SMQs to evaluate for constellations of unsolicited adverse events. No new or unexpected adverse reactions were identified based on SMQ results for either age group. Lymphadenopathy and hypersensitivity events were noted in both age groups and were previously seen in older age groups. For lymphadenopathy, there were 3 events reported from both age groups, all from vaccine recipients. And for hypersensitivity, the incidence for hypersensitivity events was actually similar between treatment groups. Most were skin and subcutaneous tissue disorders commonly seen in this age group, such as rash, eczema, atopic dermatitis, and contact dermatitis. There were no vaccine-related events of anaphylaxis reported. Here are the serious adverse events, or SAEs, for each age group. For the younger age group, 3.1% of vaccine recipients and 2.3% of placebo recipients reported SAEs. Most were common GI or respiratory illnesses or infections that occur in this age group, and none were considered related to the vaccine. For the older age group, 0.7% vaccine recipients and 0.9% of placebo recipients reported SAEs. One participant reported 2 SAEs of fever and calf pain, which were considered possibly related to the vaccine by the investigator. However, the FDA considered the events to be potentially consistent with symptoms of viral myositis. There are no notable differences found in the type, frequency, or severity of unsolicited AEs or serious AEs in either group in seropositive subjects relative to seronegative subjects, and there are no deaths reported in the study. Now we will review the Pharmacovigilance Plan for the Pfizer-BioNTech COVID-19 vaccine. The sponsor submitted a pharmacovigilance plan to monitor safety concerns that could be associated with the Pfizer-BioNTech COVID-19 vaccine. The sponsor identified anaphylaxis, myocarditis, and pericarditis as important identified risks, and vaccine-associated enhanced disease as an important potential risk. Use in pregnancy and lactation, vaccine effectiveness, and use in pediatric individuals under 6 months of age are areas the sponsor identified as missing information. The pharmacovigilance plan is for all indications as it lists use in pregnancy and lactation, which is not applicable for individuals 6 months through 4 years of age receiving the vaccine. The pharmacovigilance activities under the EUA include adverse event reporting, which may come from vaccine recipients, vaccination providers, the sponsor, or through the CDC VSafe program. Reports from vaccine recipients are voluntary. Both the sponsor and vaccine providers administering the Pfizer-BioNTech COVID-19 vaccine must report to VAERS the following information: serious adverse events irrespective of attribution to vaccination, any cases of multisystem inflammatory syndrome, cases of COVID-19 that result in hospitalization or death. Additionally, following the approval of Comirnaty, the sponsor was also asked to submit reports of myocarditis and pericarditis as 15-day reports to VAERS. The sponsor will also conduct periodic aggregate review of safety data and submit periodic safety reports at monthly intervals for FDA review. Furthermore, the sponsor has planned surveillance studies that are summarized on the next slide. The sponsor's pharmacovigilance activities also include post-authorization surveillance studies, which covers all indications for use, not just this pediatric age group. There are 4 post-authorization safety studies and 1 post-authorization vaccine effectiveness study that include individuals 6 months through 4 years of age. Study C4591009 will assess the occurrence of safety events of interest, including myocarditis and pericarditis, in the general U.S. population of all ages in the U.S. Sentinel system. Study C4591021 is being conducted in Europe and will assess whether an increased risk of pre-specified adverse events of special interest, including myocarditis and pericarditis, exists following the administration of the Pfizer-BioNTech COVID-19 vaccine. A sub-study of this study is being conducted in Europe and will describe the clinical course of myocarditis and pericarditis following administration of the Pfizer-BioNTech COVID-19 vaccine. Study C4591036 is being conducted in collaboration with the National Heart, Lung, and Blood Institute's Pediatric Heart Network, and will characterize the clinical course, risk factors, resolution, long-term sequelae, and quality of life in children and young adults under 21 years of age with acute post-vaccine myocarditis and pericarditis. Study C459-1014 is a vaccine effectiveness study being conducted at Kaiser Permanente, Southern California, that will include individuals 6 months to 4 years of age. And next, I'll go ahead and summarize the benefits and the risks for this age group. The known and potential benefits include the prevention of symptomatic COVID-19 based on successful immunobridging analyses to allow for inference of effectiveness for individuals 6 months to 4 years of age. Preliminary evidence of vaccine efficacy against COVID-19 and descriptive analyses, and the expectation of greater effectiveness against more severe COVID-19. Uncertainties in the benefits include vaccine efficacy against emerging SARS-CoV-2 variants, the long-term effects of COVID-19, the effectiveness in certain populations, and the duration of protection. The known and potential risks include reactogenicity, myocarditis, lymphadenopathy, anaphylaxis, and hypersensitivity reactions. The uncertainties and risks include the safety in certain populations and adverse events that are uncommon or that require longer follow-up to be detected. And here I will end with our voting question for our committee members today. Based on the totality of scientific evidence available, do the benefits of the Pfizer-BioNTech COVID-19 vaccine, when administered as a 3-dose series, 3 micrograms each dose, outweigh its risks for use in infants and children 6 months to 4 years of age? Thanks so much.
Thank you, Dr. Wollersheim. Very succinct and careful presentation. Dr. Cohen, you've got your hand raised, followed by Dr. Bernstein.
Thanks, Dr. Wollersheim. That was really an incredible presentation. I really appreciate the transparency and clarity. 2 questions. One is it possible for FDA, or did you tease out reactogenicity in the population of infants and young children who got vaccinated with that 3rd dose between 8 and 12 weeks? And is it similar to the kids who got vaccinated more towards the median 3rd dose interval that was done in the study? And then my second question is, when you is the FDA assessment that the 3-dose primary series of Pfizer is based on immunobridging studies meets the criteria compared to 2 doses of adult vaccine, or are you able to also feel confident that it would be similar to 3 doses in the adult vaccine?
Thanks, Dr. Cohen, for great questions as always. Your first question was related to reactogenicity based on the dose interval between dose 2 and dose 3, if I understand the question. And we did try to do that analysis. And from what I recall from that, I'm sorry I don't have a slide to show you with those numbers, there was no significant difference in the reactogenicity based on dose interval. And your second question, I think, is related to comparisons of vaccine effectiveness following 2 doses in adults or 3 doses of adults. Is that correct?
Yes.
Okay, thank you. And I think that's a difficult question to address based on the data that we have here, because our immunogenicity comparisons are to adults who received the 2-dose primary series. There's further discussion in some of the benefit-risk profile and within our briefing document with the benefit of the 3rd dose for adults. And so I think that benefit is really reflected by the variant that's circulating currently, the Omicron variant. And I don't know that the data would be available from the appropriate time periods to make that comparison at this point, if that makes sense, because we don't have data pre-Omicron for these younger age groups.
Great. So is FDA's assessment, though, that there would also need to be an additional dose, a booster dose in this population?
That's a great question. Thank you. And I might defer to Dr. Fink if he wants to weigh in here, because it's a bit beyond the scope of the data that I presented from the study here. Thank you.
Yeah, thanks, Susan. Happy to weigh in. I think we have a situation here where, as you've seen from the Pfizer presentation and FDA's independent analysis of the data, we have some very preliminary vaccine efficacy results after dose 3 that are limited by a small number of cases and limited follow-up time that appear to suggest a improvement in protection following dose 3, as compared to following dose 2. We do consider this estimate to be preliminary. We consider it to be imprecise and potentially unstable. And so exactly what the vaccine efficacy is after dose 3, I think needs further data to inform. And we would expect to get some of these data, hopefully from updated analyses from the clinical trial, if more cases are accrued, recognizing, of course, that if the vaccine is authorized, that will result in unblinding of placebo recipients so that they can get their 3-dose series, and also from real-world effectiveness data once the vaccine is used. I do want to make it very clear that based on the totality of evidence that we've presented, including primarily the immunobridging data to the 2-dose adult primary series, as well as a number of pieces of supportive data, including preliminary descriptive efficacy analyses and other inferential lines of data that you've seen from Pfizer, that we do feel very confident that the evidentiary standard for benefit for EUA has been met here. But I think in terms of what the efficacy is after a 3rd dose and whether an additional dose beyond that would be needed is going to require a little bit more data to sort out. Thank you.
Thank you so much.
Thank you, Dr. Fink. We're moving on. 2 more questions. We have a hard stop, or 1 more question from Dr. Offit. Dr. Bernstein, you're next.
Thanks.
I'm juggling things here.
Great presentation, Dr. Wallersheim. And I just had a question. Could you go to slide 27, please?
Which one, sorry?
With the Omicron neutralization analysis that was done, they used a fluorescent focus reduction neutralization test. And on slide 27, it shows, I think it's a slide before that. Yeah, on that slide, it shows that the post-dose 3 GMTs against Omicron were lower as compared to the ancestral strain and the Delta variant. What's the clinical significance of that?
Thank you for the question. And I think that is a great question because we're not sure. We don't have a correlate of protection. Additionally, this is a non-validated assay that's been used just to have this information so that we can see that there is neutralization of these various viruses. The clinical interpretation, though, I think is limited at this point in terms of what my understanding of what this assay shows. I welcome Dr. Fink if he has additional insights to interpretation of these numbers as well.
Thank you. I think what we can say is that the data you see on the slide here tracks with what we've seen in real-world effectiveness studies and immunogenicity studies of this vaccine and other vaccines in older age groups, that the neutralizing antibody titers are lower for Omicron compared to Delta and the ancestral strain, and that does correlate with a lower level of effectiveness against at least more mild disease, and in some cases more severe disease due to Omicron than the Delta variant and ancestral strain.
Okay,
but I don't think we can conclude anything specific based on these titers.
Okay, Dr. Offit, I'm trying to get our organizers the time to prepare for the OPH. Dr. Offit, please.
Okay, so Dr. Wollersheim, again, thank you for that clear presentation. What I'm trying to understand, if you look back in December 2020, when we considered the Pfizer and Moderna vaccines as two-dose vaccines, they were essentially equivalent in terms of efficacy. Similarly, if you look at the efficacy for two doses of Pfizer versus Moderna for the 12 to 17-year-old or the 6 to 11-year-old, again, they tracked as being similar. This is the first time you're really seeing a difference in the less than 5-year-old, where two doses of Moderna does offer some level of protection, whereas that wasn't true at all for the Pfizer vaccine. And the challenge strains were essentially identical, which is the Omicron era. And I realize these weren't side-to-side comparisons, but do you have any sense of why that would be true, why there was, for the first time, a clear difference where Pfizer's didn't protect after two doses, where Moderna had, where they had tracked so similarly previously?
Thanks, Dr. Offit. Great question, of course, and I don't want to say impossible, but it's difficult really to make the comparisons between these two vaccines. So I would, I don't think we can really move there with the data that we have. But thank you for the question.
And we can revisit that question later on because I'm sure it's going to come up again. Thank you to all our presenters. It's now time for a half-hour break. We will resume at 1 p.m. Eastern for the oral public hearings, the OPH.
All right, thank you, Arnold. And yes, let's take us to break. Please hold off, members, for a moment before we go to break. Studio, please put us on break.